Hello, everyone, and thanks for joining us for day two of the Advances in Cancer Therapies and Diagnostics Symposium. I'm Nathan Nee, Senior Science Editor for The Scientist, and I will be moderating our discussion. Today, our expert panel will discuss basic preclinical and clinical cancer research involving new diagnostics and therapeutics. Before we begin, I'd like to thank Epi, Cypher, Biorad, Sinobiological, Biodium, BD Biosciences, BioXcel, Scale Biosciences, and Selecta for sponsoring today's webinar. We'd also like to extend a very special thank you to our platinum sponsor, Biomodal. We like our symposia to be interactive, and we encourage you to send us your questions or comments at any point, and each speaker will address relevant questions during the Q&A session that follows their individual presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsors have provided us with some helpful resources related to today's webinar, and we have posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. If you have minimized the Q&A panel, please simply click on the blue chat button in the bottom right corner to bring it back. This symposium will be archived on the scientist's website, and we will send you the on-demand version link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our first speaker today. David Liu is a medical oncologist and computational biologist at the Dana-Farber Cancer Institute and Brigham and Women's Hospital, with a background in computer science, biostatistics, and epidemiology. Prior to entering medicine, he was a software engineer and analyst at Amazon.com in the personalizations group. He runs a computational laboratory at the intersection of medical oncology, computational biology, and molecular oncology, developing computational approaches to dissect and predict response and resistance to cancer therapies, integrating molecular and clinical data from patient samples. Liu's research focuses on discovering biologically relevant molecular connections between treatment response and resistance for novel therapeutic targets and rational combination therapies and building clinically relevant predictive therapeutic models. Dr. Liu, let's just make sure everything is up and running from a technical perspective. So if you would please share your screen.
Hello, can you guys hear me?
Yes, we can hear you and I can see your slides. Please, the floor is all yours.
Great, thank you so much. So thank you for the kind introduction. Thank you to the organizers of this webinar for the opportunity to speak here. My name is David Liu. As mentioned, I'm a medical oncologist. My focus is in melanoma. I'm at the Dana-Farber Cancer Institute, and I run a computational lab here as well. So today I'll be talking about some of our work about learning from patients, molecular biomarker discovery, and tumor evolutionary dynamics. And I'm just going to set a timer here so I know that I'm going to run out of time. No conflicts of interest. As mentioned, my background was previously in computer science and economics. I was at Amazon.com as a software engineer and an analyst within the personalizations group, a period of time before going into medicine. After going into medicine, I actually did my fellowship at up here in Boston at the Dana-Farber, where I kind of fell into this area where I actually was able to combine these different aspects of clinical medicine, of computational approaches, and cancer biology. So, which is what I do now. So the kind of work and the kind of that my lab is interested in is kind of to understand what the drivers of clinical heterogeneity are. So really taking what we observe clinically is that there's a huge variability and heterogeneity in terms of patient outcomes and response. For instance, response to different types of treatment, from rapid progression and intrinsic resistance to durable responses and long-term durable responses. Other different types of phenotypes, clinical phenotypes, is that patients have very, even within the same histology, they have very varied patterns of metastases, which are not well understood. But to better characterize those, the different patterns of metastases, as well as their molecular drivers, as well as kind of doing these evolutionary studies about, so deep dives into individual patients to basically understand within the context of their clinical history what the course of tumor evolution is over progression, metastasis, and development of resistance to therapy. And the overall arching of all of this is that this is all driven by differences in molecular characteristics and the biology of tumors, even within the same cancer type, between patients as well as between tumors, which is inter-patient tumor heterogeneity, as well as tumors within the same patient, as well as even within the same tumor, that there's heterogeneity within the tumor. So I have three kind of stories that I'll talk about today, and there's a lot more in the papers that underpin these stories, but I'll try to pick and choose from different aspects here, and hopefully you find this interesting and informative. So the first story here is about developing predictive markers, biomarkers of response to anti-PD-1 immune checkpoint blockade. So in advanced melanoma, there's actually, so immune checkpoint blockade has really revolutionized the treatment and prognosis of cancers, is broadly used now across a range of different cancers. Melanoma was actually the cancer type where it was first demonstrated to be effective, and it still remains to be the cancer type that where it has the greatest impact. So, but now in the first line setting, there's kind of this clinical question and conundrum, which is there's a range of different immunotherapy regimens which are approved in the first line setting, and how do you choose that for the patient who's sitting in front of you? So starting with single agent anti-PD-1, where the response rates are 30 to 40%, and then going down to the combinations, where the combination anti-PD-1, anti-CTLA-4 at the original regimen has a 51% response rate, but the issue is that along with the increase in response rates, you also have a huge increase in toxicity and severe toxicities that occur, up to 50% for the combination versus 10% for the single agent. And this is actually the randomized controlled trial CheckMate 067, where this is the 10-year follow-up data, where you can kind of say nivolumab is the anti-PD-1, the green arm here. I hope you guys can see my cursor. Whereas the combination anti-PD-1 CTLA-4 is seen here. And what you see is that the survival certainly does appear to separate, right, between single agent anti-PD-1 nivolumab versus the combination. But the absolute magnitude of the difference, if you look across it, at 10-year estimate is about 6% absolute difference. At the 5 years, about 8% absolute difference, which basically means that, and those are the extremes here, right? So, but that there's only a 6 to 8% absolute difference in survival between anti-PD-1 combo of therapy, but the combo therapy is actually is much more toxic compared to the single agent. So it raises the question: can we identify which patients are likely to be resistant to single agent anti-PD-1 and actually would in fact differentially benefit from combination therapy? And this is a question that we wanted to address here. So this is in collaboration between the Dana-Farber and our collaborators at University Clinic Essen, so Dirk Schaddendorf and my co-first author, Bastian Schilling, where we created a cohort of metastatic melanomas that were treated with anti-PD-1 immune checkpoint blockade, where we had pretreatment samples and did whole exome sequencing and bulk RNA-seq, along with very deep clinical annotation. The data that was generated for this particular study was from the whole exome sequencing, so the somatic mutations that were associated with each individual tumor, as well as overall genomic characteristics such as the tumor purity proportion of the tumor sample that is actually from tumor cells, genomic ploidy, genomic heterogeneity, as well as mutational signatures. From the bulk transcriptomics, we inferred different aspects of gene sets, hallmark pathways, characteristics of the immune microenvironment, as well as antigen presentation. And the clinical data is all the response data to therapies, PFS-OS, as well as prior therapies and different lab settings. And so the first hypothesis I want to share with you here is at the time there's a question, and even now, that hey, what are predictors of response to immune checkpoint blockade? So the way that immune checkpoint anti-PD-1 is it blocks a it blocks it blocks a receptor that downregulates the immune response, and so blocking the blocker upregulates the immune response. So the idea is that if you have immune infiltration of the tumor, but that is an ineffective immune infiltration, that blocking anti-PD-1 restores the anti-tumor phenotype, and therefore there's an effective response. So that the hypothesis is that the immune infiltration predicts response. And actually, what we found instead was actually something a little surprising to us and actually nuanced here, which is overall within this reasonably sized cohort. So what we did was we split the cohort into high and low immune high and immune low scores. And we found that there was a little bit of a difference, right? So the proportion green here is responders in the higher versus the low, but it wasn't really all that significant. It didn't seem like a very good biomarker. However, when we then went back and overlaid this question of whether they had been previously treated with immune checkpoint blockade. So recall they were treated with, in this study, anti-PD-1, but about half of the patients previously had been treated with IPI with anti-CTLA-4 because that was the standard of care at an earlier time point. And what we found was that this difference in terms of immune infiltrate, in terms of responders and non-responders, was really only in those who had a prior history of being treated with immune checkpoint blockade with anti-CTLA-4. But in those that were naive to prior immune checkpoint blockade, there was no difference at all. And so this led to this model or this understanding where if you had previous treatment with anti-CTLA-4, that's ipilimumab, but when you biopsied after that, after progression, and you saw that it was a cold tumor, there was no immune response, that predicted very strongly that you were not going to have subsequent response to anti-PD-1 immune checkpoint blockade. Whereas for the other combinations where you had a significant immune infiltration post IPI progression, that you actually could respond, or you could actually progress. And similarly, in the IPI naive setting, so whether or not you see an immune cold or immune hot environment, you could actually see a response or resistance. So it does not seem to predict that. So subsequently, what we were interested in doing, seeing this very large difference between the two, we hypothesized that we should really change the way that we approach the modeling, because the predictors of response were so different between whether or not you had prior treatment with immune checkpoint blockade. So then we built separate models for these two different settings. And what we did was we integrated clinical genomic and transcriptomic features too. And we wanted to build the parsimonious predictive models of response in integrating all this data. And so in the IPI experienced tumors, what we actually found, and the reason for the parsimony was because we wanted to not overfit for one and also have a more interpretable model. And what we found was in the IPI experienced tumors, combination of clinical features and molecular immune features actually predicted pretty good, was actually quite a good predictor of intrinsic resistance in the setting. So MHC class 2 is antigen presentation, very strongly related to kind of the immune response or immune infiltrate within the tumor. LDH is a marker of tumor burden, and the presence of lymph node mets is a clinical feature about the presence of met sites. But put those things together, and you have a pretty good predictor of AUC within this particular setting of prior IPI, and you see the differences in PFS and OS between kind of median split in this population. So, and on the other hand, those that are IPI naive, so those who have not had immune checkpoint blockade, a very different set of features actually was predicted intrinsic resistance. So genomic heterogeneity, high genomic heterogeneity, low purity predicted primary resistance to, and purity predicted resistance to anti-PD-1 in the IPI naive tumors. And you see the separations here. And this is actually the more clinically relevant setting because, as I mentioned, anti-CTLA-4 is not first-line therapy anymore. And so basically all the subsequent patients who were looking at this decision would be more in the IPI naive or immune checkpoint naive setting. So the idea here, the concept is that prior therapy matters, right? So ipilimumab treated in naive melanomas actually have different predictive features. And this was actually subsequently reproduced by others in independent data sets and in meta-analyses, for instance, the group at UCLA, Tony Rubop's group just last year. And so we developed these parsimonious predictive models separately for IPI-naive and IPI-treated patients. And then these genomic features were the ones that predicted anti-PD-1 response. At the time that we published this paper, we can only perform limited validation in independent cohorts because the number of cohorts that actually had all this information, clinical whole exome sequencing and transcriptomic information, was basically very, very, very few. Subsequently, we were able to partner with some industry partners, so get information from some clinical trials as well as some additional cohorts that had whole exome sequencing as well as IO treatment and response to therapy. And we were actually able to reproduce, again, focusing on the IPI-naive setting, that high genomic heterogeneity and low ploidy were robustly and strongly associated with intrinsic resistance to anti-PD-1 immune checkpoint blockade in 3 independent cohorts. So really, this seems to be something, and this work was led by a postdoc in my group, Dr. Giuseppe Tarantino, and published this past year. We then went on from that, so genomic heterogeneity and ploidy, to basically say, instead of just saying, hey, these are robustly significantly different between responders and non-responders, can we do something a little bit better? So the clinical question is, if we can robustly identify a set of patients with high confidence that they're going to be intrinsically resistant to single-agent anti-PD-1, then these are the patients that we can confidently say they would differentially benefit from the combination therapy, and therefore the toxicity benefit ratios would be better in those patients. And so what we did here was we tweaked the model, the weighting of predictions in the model, so the error calculation in the model, to favor the development of a model that has high positive predictive value. So you may not identify every patient that's intrinsically resistant, but the ones that you predict you're very confident about. And we found this particular model here, which is a simple decision tree, had a odds ratio of almost 17 and a 90% absolute positive predictive value. And this is the survival curves that you see here. And that was just in the training data. We were able to find another separate cohort. Granted, it's a relatively small cohort, but of anti-PD-1 treated patients. And what you see here is in this little orange area that it actually predicted 5 out of 5 patients correctly predicted intrinsically resistance. Then the question is, for those same patients who fit in those same criteria, if you treated them with combination anti-PD-1 CTLA-4, would they actually respond to therapy? And so there's another cohort that we have, again, small, but treated with anti-PD-1 CTLA-4, that were predicted as 3 out of 7 patients that would predict as intrinsically resistant to single agent actually respond to combo therapy. So this is kind of proof of principle here. So heterogeneous employee robust predictors across multiple independent cohorts, and we built this optimized predictive model to address directly the clinical question of identifying patients with high confidence that are going to be resistant to single agent who would benefit from the combination therapy. Moving forward, these features that we have identified are from whole exome sequencing, which is not routinely clinically done, and also the pipelines for analysis are not as standardized kind of across centers. So what we're interested in doing is actually taking clinical assays. So we hypothesize that some of these features, genomic heterogeneity as well as ploidy, may actually be inferable from pathology images. And pathology images are all, H&E images are present for every patient, every cancer patient. And using some of these advances in AI and foundation models for pathology images, basically to predict ploidy as a way of very rapidly getting to clinical translation of these potentially predictive features, both for validation and for prospective studies. And we're also interested in the biology of this, which is to understand because these heterogeneity and ploidy are not mechanisms in and of themselves, but they are means to a mechanism of developing, particular mechanisms of resistance and response. So doing single-cell RNA-seq type studies to try to understand what the correlates of heterogeneity and the microenvironmental features associated with both of these particular features. Okay. Second story that I wanted to talk follows out of the original this observation as well. So based on the prior, the differences in the different immunotherapies, the different response rates, the different toxicity rates, one of the questions we wanted to ask is, can we pull, now with more data, can we identify mechanisms of resistance to different immunotherapy regimens and targets to improve the efficacy and reduce toxicity? This was work led by Amy Huang, who is a PhD student at MIT. Can we identify which patients are likely to have specific mechanisms of resistance? So more a biomarker-driven to identify mechanisms of resistance and would benefit, therefore, from specific combinations. And so in order to do this, what Amy did was she pulled together all the data that had been published where we had, in this case, bulk RNA-seq that was taken from of melanoma, advanced melanoma patients treated with some form of immune checkpoint blockade, where we had good clinical annotation. And so Amy went through and very carefully annotated specifically cutaneous melanomas, known cancer types, as well as good quality bulk transcriptomic data, and specifically, the response data, and then broke them down into patients who were treated with one of 4 different therapies: anti-CTLA-4, anti-PD-1 with prior anti-CTLA-4, and the combo therapy. And so one of the first things she looked at was to say, well, let's look at the immune microenvironment with this idea of maybe there are differences that in the immune microenvironment between responders, and it may be informative. Well, it turns out that the set of signatures, which was first showed by Bagayev, we didn't feel confident that they were very strongly correlated with each other, and we didn't feel confident that we could distinguish between specific tumor microenvironments in terms of deconvolution of the bulk RNA-seq. But what we did see is that the cohort kind of split out into these 2 groups of immune high and immune low groups. So grossly speaking, those with high immune infiltrate and those with low immune infiltrate within the tumor. And so the hypothesis that we took forward was that the biology and biomarkers of response to resistance to immune checkpoint blockade are different in immune high versus low tumors. So remember that in both these cases, there are responders and non-responders to therapy in both the immune high and the immune low group. And so the idea is like the biology of response to resistance may actually be different in this particular setting. So the idea is that stratified analysis of immune high versus low responders and non-responders and would actually reveal generate more homogeneous cohorts, where then you have better signal to noise ratio to be able to see this. And there's more in the paper, but just focusing on a couple of things here. So in immune high tumors, what we found was that hypoxia-related pathways were identified as associated with resistance to single-agent immune checkpoint blockade. So we found that some of the strongest pathways were upregulated in resistance to a single-agent anti-PD-1, specifically within immune high tumors. So hypoxic glycolysis, as well as EMT, and not statistically significant VEGF and angiogenesis. And this just shows it here, where you can kind of see in the immune high tumors, specifically, there's a higher hypoxia response score in the non-responders, specifically in the immune high setting, but not in the immune low setting. And hypoxia is, of course, there's a large body of literature that has been done primarily in preclinical settings that has demonstrated the effect of hypoxia on immunosuppressive cells and states. So macrophage polarization towards a more pro-tumor type phenotype, upregulation, recruitment, and upregulation of Tregs, as well as immunosuppressive effects on antigen presentation in DCs, NK cells, and cytotoxicity in CD8 T cells, exhaustion, etc. So this is well known that this is likely to have an impact on the immune microenvironment. And usually, and the way it works is through these a pathway, HIF1 alpha, HIF1 beta, as well as some overlap with HIF2 alpha as well. But basically, hypoxia induces these changes where typically in a normoxia setting, you have sequestration and rapid breakdown of the HIF1 alpha beta complex. But in hypoxia setting, that they persist and they then downstream activate a number of pathways that then leads to changes in the tumor cell metabolism, angiogenesis signals, invasion, metastasis, and ultimately survival. So what we were interested in then from this is then to understand, to try to look within clinical tumor samples, what the correlates were of a hypoxic tumor microenvironment. And so for this, we worked with Jackie Yang, who is now started her own lab at Rutgers. PI was Melose Callis at MIT, and in collaboration with Jean-Viet Bolland at MGH, where we had a single cell cohort of immune checkpoint blockade treated patients with pre-ICV samples, where we had a single-cell RNA-seq. So one of the first things we saw was there's a range of hypoxia that we saw, right? So across this cohort, we calculated a sample hypoxia score and then generated and saw, there's a range of different overall hypoxia. And then when we looked at what the range of hypoxia was associated with, we saw that greater hypoxia overall sample scores was associated with lower immune infiltrate. Not surprising, though there is some variability, as you can see, between low, mid, and high, but also immunosuppressed phenotypes. So within correlated with more hypoxic microenvironments and scores are markers in CD8 T cells, as well as macrophages, that are associated with more exhausted or less cytotoxic phenotypes. What was interesting, though, was when we looked across different cell types here, is that stromal cells, so in this case, fibroblasts and endothelial cells, actually have the highest expression of different aspects of hypoxia signaling. So, many cells express HIF1 alpha, but HIF2 alpha was specifically expressed in fibroblasts and endothelial cells. And then the VEGF receptors, also VEGFR1 and EGFR2, which is FLT1 and KDR, are also strongly expressed in the endothelial cells. And moreover, what we saw was that when we looked at ligand receptor interactions and relationship with hypoxia scores, what we saw was that there was a pretty strong relationship between the number of fibroblast tumor interactions with tumor stromal interactions with the increase in a hypoxia score. So suggesting that with hypoxia, there is actually that the stromal compartment is an integral kind of part of a mediator of the effect of hypoxia. So this led to this hypothesis that maybe we can try to modulate the hypoxia pathways in immunogenic tumors, right? To help overcome immune checkpoint blockade resistance. And in particular, we thought that it would be interesting to try to target HIF2 alpha, right, which was specifically expressed within the stromal cells. So we then took this back to a preclinical model, and this is work that was led by Kelly Berg, a talented postdoc within Arlene Sharp's lab at HMS, and looked at syngeneic melanoma cell lines within immunocompetent mice with either low or moderate response to immune checkpoint blockade with and without HIF2 alpha inhibitor and anti-PD-1 immune checkpoint blockade. So this is just kind of the experimental schematic where you have flank injections in these immunocompetent mice, and then subsequent let them grow out, and then subsequent treat them with a 2x2 of either anti-PD-1 or control or a HIF2 alpha inhibitor or control. And the two melanoma models that were tested were basically B16, which is known for having low immunogenicity, so more of an immune cold setting and not typically responsive to immune checkpoint blockade. And then B16 ova, which is kind of a modification of B16 that has moderate immunogenicity and moderate responsiveness to immune checkpoint blockade. And what we see here is that in the B16, so the idea is B16 ova is more of the immune high setting that we see. And the hypothesis that indeed that we see that the HIF2 alpha plus anti-PD-1, and that's this gold curve here, that combined improves tumor control and moderately immunogenic. But when we look at the B16, that was immune cold setting, we don't see this effect again. That's gold. So findings, kind of proof of principle that there is this combination effect within the immune high setting. And so the summary of this work is basically, through these stratified analyses of the immune high and the immune low tumors, we have identified these hypoxia markers, right, as these biomarkers of single agent anti-PD-1 resistance and potential target, and a potential target specifically and differentially in the immune high tumors. So we found that these greater hypoxia within clinical samples is associated with this decreased immune infiltrate and more immunosuppressive microenvironment and phenotype, as no surprise. As we would expect, but it's also associated with increased tumor stromal receptor ligand interactions, which was kind of an interesting. And so we thought targeting that a hypoxia modulator within the stromal compartment might actually be a synergistic effect with anti-PD-1 and have some functional data that shows that proof of principle in this. So the implications of this work is clinically, immune high tumors might actually be a biomarker here, right? They may disproportionately benefit from combining anti-hypoxia angiogenesis agents with anti-PD-1 checkpoint blockade. And in fact, there are ongoing or completed or nearly completed trials of combination anti-hypoxia or angiogenesis agents plus anti-PD-1, but kind of in the immunotherapy progressive melanomas who have had prior immunotherapy. So we are interested in testing whether immune high tumors predict patients who actually benefit from this combination of therapy. Other things that's similar to prior is that developing a clinical assay of immune high. What we inferred from here was immune high was from the bulk RNA-seq setting, but coming up with a clinical assay from H&E, et cetera, as well here. And in particular, I just wanted to point out that this is kind of innovation in biomarker discovery. So rather than just simply saying responders versus non-responders kind of in an unselected way and saying what are the differences, the suggestion here is that if we can do stratification of patient subgroups in like a biologically relevant manner, right? So the immune high versus immune low, then we could probably sharpen the signal to noise ratio, or I would argue that there are different mechanisms of resistance that are likely happening within those different subgroups. And by separating them out, we can actually more cleanly and identify particular signals. And of course, biology, substance coordinators was the mechanism of improved tumor control within these combination treated immune high tumors, which is ongoing as well. Okay, third story, and I'm going to try to run through this, is a tumor evolution story, the power of one. All right, so this is in collaboration with Peter Sorda's group, Laboratory for Systems Pharmacology, and Jerry Lin, who is a senior scientist within his lab, and along with Jean-Vive Bouland, who really led a lot of this work. And this was published in 2021. This is a deep dive into an individual patient. So the clinical history of this patient: 67-year-old man. He presented with a localized high-risk disease, so on his back, that was discovered on routine physical, and he was treated appropriately with a wide local excision and sentinel lymph node biopsies, which did not show any metastatic disease. He was then followed appropriately, and then 2.5 years later, unfortunately, he presented with eruption of subcutaneous nodules in his left forearm and chest wall, which were proven to be metastatic melanoma. Restaging scans, you see the PET CT images here. All the little dots here that are not the heart, kidneys, bladder, or brain are metastatic lesions. So you see lots of little dots through here. So he had widespread metastatic disease at initial presentation. He was put on a clinical trial of sequential immune checkpoint blockade. He was first treated with anti-PD-1, where he had no clinical response, and he had rapidly growing disease in many different places, ulcerating lesions, bad disease progression. Then switched on trial to ipilimumab and continued to be resistant and progress. But then somewhere between his second and third cycle had this almost Lazarus-like effect where he started responding and all the lesions disappeared. And so he clinically had a complete response and subsequently went on to complete 2 years of maintenance immune checkpoint blockade with no recurrence of disease and no progression, which is great. About 10 and 15 months after completing the 2 years of maintenance therapy, he had a development of single METs that were progressive detected on scans and that were treated locally. He had a small bowel MET initially discovered that was resected, and then he had a brain MET that was discovered, resected, and radiated. With no subsequent systemic therapy for another 1.5 years, he then unfortunately developed widespread progression that was refractory to all subsequent therapies and ultimately died from his disease. So from this one patient, we ended up collecting 37 tumor samples collected from pre, on, and post-treatment time points over 9 years of time. And so this was from so we did whole exome sequencing, bulk RNA-seq, and SciSef, which is a multiplex IF on this patient. And we did phylogenetic analysis on the patterns of somatic mutations that were in this. And I'm happy to go through, exactly how we did this phylogenetic analysis. But basically, the phylogenetic analysis of these 37 tumors, and then overlaid that on top of the clinical history. So what we see here, each dot here, each line color represents a different lineage. The dot represents a different lesion that comes from a different location. And so what we see overall is that all the tumor samples ultimately came from a common ancestor that had hotspot mutation in MAP2K1 and IDH1. But each of the different lineages, however, they popped out of that, had a distinguishing set of mutations. So copy number alterations, as well as a cluster of mutations that distinguished that lineage from another. And during that initial period of progressive disease, what you see is continued propagation of these 7 different lineages over that period of progression. So in parallel on the same patient. After the response, all the lineages disappeared, but all the subsequent resistant lesions that popped up from the oligomets to the ultimately fatal downstream widespread metastases all arose out of a single lineage, which is the green lineage here, that was characterized by a chromosome 15q loss. So the first question that we're asking is, why did the first resistant clone arise from lineage 3? And so 15q, loss of heterozygosity, it encodes for a particular region, a gene, beta-2 microglobulin, which is necessary for MHC class 1 presentation. So loss of beta-2 microglobulin expression leads to essentially evasion of T cells and recognition from T cells. And so the idea here is that the lineage 3 tumors had a decreased expression of this, and maybe as a way for them to hide from this. Another possibility is that the loss of heterozygosity was a large portion of the 15q chromosome that was lost, including a large number of mutations that were present there. So if there was an immunogenic mutation that was actually present there, loss of it could actually also lead to immune evasion. Other evolutionary dynamics of resistant lesions in lineage 3 didn't just stay static over time. So when you went from the early lesions that disappeared with immune checkpoint blockade to the later lesions that reappeared, they actually acquired additional drivers and additional genomic events. So resistant lesions in lineage 3, they developed genome doubling type events and acquired CDKN2A biallelic deletion that you can kind of see here. Whole genome doubling is actually associated with increased aneuploidy or alterations within the chromal structure, so copy number gains and losses within the tumor, which allows the tumor to actually be able to persist and survive aberrations, but allows it to explore the space of alterations and potentially find resistant and growth phenotypes. So, and this is basically just showing the proportion of genome with aneuploidy or copy number gains and losses, and genome doubled have far higher proportion of the genome with those compared to non-genome doubled tumors. This is within the same patient. Other folks have basically shown that when you follow patients over time and they develop resistance, that aneuploidy seems to be a continued ultimate destination in some ways, that you have increased aneuploidy as you kind of go down the line of resistance. And the late resistant tumors in this particular patient also continue to evolve, right? So even from the resistant tumors that popped up to the later fatal disease, there continued to be continued alterations and loss. Late resistant tumors had increased aneuploidy even compared to the early genome double tumors. And other observations from here is that the brain metastasis, this R2 that you see down here, that was resected, interestingly, was an evolutionary dead end. No subsequent resistant lesions were descendants, including 2 brain mets that were seen at rapid autopsy. Instead, those actually arose out of this R1, the small bowel lesion. So it suggests that actually this definitive resection radiation was effective in eradicating this brain clone. So, to say, clinical tumor heterogeneity evolution can be incredibly complex and nuanced. One of the really important lessons, at least for me, in terms of understanding what's happening with this patient, is that it changed the way I thought about immune evasion, because usually you have this idea that, hey, if you have a resistance mechanism that you find, then you're going to be resistant to you're going to evade the immune system, you're going to be resistant to immune checkpoint blockade. But actually, the paradigm should be that the tumor has to evade the immune system in order to become clinically evident. And so it's really more about the tumor and immune system are kind of in an arms race. And this has the implication that basically no single tumor intrinsic immune evasion feature is likely to be definitive, right, in predicting IO response, because it is this arms race. And what we see, as in this case, that resistant lesions accumulate multiple resistance-associated alterations. Despite the crazy heterogeneity that you see, immunotherapy was still extraordinarily effective in eradicating most of the clones, but the disease continued to evolve after development of resistant disease. And this genome doubling probably allowed increased aneuploidy that may cause changes in the functional state and the development of resistant clones. So implications of this, the idea here is that the steep molecular characterization of longitudinal tumor samples from patients across time, treatment, metastatic sites actually, I think, gives us really important insight into clinical tumor evolution and the drivers of metastasis and therapy resistance. So they directly elucidate kind of this heterogeneity and the mechanisms of patient disease trajectories, where you see changes from before and after, and the earlier ones are like controls, right, internal controls to see what changed, and those are potential mechanisms. The downside, of course, is the generality of these specific findings are not clear. But how many patients does this apply to? Is this a general rule, or is this a thing? And it really requires us to do much more of these types of studies to basically understand what the common pathways are. But the lessons about clinical tumor evolutionary dynamics, in my opinion, cannot be learned any other way. And these are the relevant things. So the last thing I just wanted to close with is just some thoughts around lessons around doing this type of work, clinical computational oncology, that I hope could be helpful. One is that dissecting clinical heterogeneity or learning from our patients is actually hard. So one of the most important frameworks I feel like that I've learned from this is that there are many paths to Rome. So the mechanisms of resistance, the types of resistance, the types of response, the types of these different phenotypes are actually heterogeneous. It's not that you have a single mechanism or things that lead to a particular phenotype. And in the vast majority of cases, any individual mechanism is probably relevant in just a subset of patients. And so what the implication is that we really need large cohorts for statistical power, right, to be able to detect these things in ways that we feel are robust. And direct problem with that, patient samples are hard to obtain, they're variable in quality, and they're challenging to analyze. So varying from small needle biopsies that may not do a good job of sampling the tumor versus larger section samples. There's a limited sample size at any individual institution to be able to put together cohorts. Tissue processing, like formalin fixation, limits what kind of molecular assays are possible and the quality of the data. Also, how long things sit out before you process them make a huge difference. There are many confounders, so biopsy site, age, sex, and heterogeneity in the clinical context, which also make it very, very hard to analyze the data and generate robust conclusions. Generating the multimodal molecular and clinical data sets is hard and it's expensive, right? You look at that patient and you think about how much had to go into that in order to generate this map. And detailed clinical annotations, like of all the sources I was talking about, are critical, right? But they're often not available in molecularly characterized cohorts. Now, one place where this is available is large cohorts of these clinical homogeneous and treated patients with standardized annotations of treatments and outcomes and everything else. They're in clinical trials. But unfortunately for the field, this data is often locked up and very, very difficult to obtain. However, it is important. Despite all these challenges, the biology underpinning this clinical heterogeneity is the important biology to learn, right? Because this is what's actually driving disease phenotypes. And the only way, in my opinion, to understand clinical heterogeneity is to actually study clinical heterogeneity in patients. Now, the limitations of this, right, is that the findings will most often, they'll be correlative and hypothesis generating and not causal, right? Because we're not doing the experiments in patients, and that is a limitation. Cohort sizes will almost always be too small to find statistically significant results, especially when you correct for multiple hypotheses. Many paths will lead to Rome. But in that particular setting, you really have to apply your biological knowledge to prioritize what you think are plausible hypotheses, and also to think about what the corroborative experiments and data might be to verify that this is indeed an important finding, or it is a real finding, not just by random chance. And a lot of healthy skepticism towards any finding. Nominated findings, you need validation in independent cohorts. And also the problem is that you oftentimes have variability in different cohorts that lead to non-robustness or non-validation within these cohorts. But if you're lucky, the cohorts will vary in ways that are actually measurable, so you can figure out what the differences between the cohorts actually are. And obviously, this is very complementary to the preclinical work that our colleagues do, functional validation necessary to prove causality and dissect mechanisms and potential targets for therapy. So interdisciplinary collaboration is key. Working with our colleagues for basic translational scientists in terms of their biological knowledge, functional validation, mechanistic dissection, and working with our clinicians, doing these integrated molecular analyses of clinical cohorts and generate the samples and the data, clinical phenotypes of interest, and synergistic expertise with our computational colleagues. A computational biologist does not do all things, and there's certainly expertise in many different ways that we can leverage in collaborations and combinations. So with that, I will thank, first and foremost, the patients whose generosity and willingness to be part of these studies have made them really, really possible and motivate everything that we do. Members of my lab who have stuck through with me and really, generated the data and the studies that you see here. Lots of collaborators from across Dana-Farber, MGB, Broad, Essen, Harvard Medical School, and my funding sources. And I thank you all for your attention and also for caring about science, which feels like it's challenging at the present time. So that's all. Thank you.
Thank you very much, Dr. Loop. So we'll just move on quickly to our Q&A session. And I personally wanted to ask, especially kind of in light of how you concluded the talk, with clinical heterogeneity being what it is, and it feels like the more we learn about it, the bigger, I don't want to say problem, but things become more heterogeneity, things become more heterogeneous.
Yeah.
So with that, is generality, is the pursuit of finding something that could be used across a broad population or a broad swath of the population, is that becoming less realistic in terms of dedicating resources for that goal in looking for something that may not actually exist because of heterogeneity?
That's a really important and that's a really good question. So I think there's been paradigm changes, kind of, so immunotherapy or immune checkpoint blockade, I should say, right, in the last 15 years. The mechanism of it is not targeting the tumor cell at all, right? It's about taking the brakes off the immune system and somehow magically, and I still feel like it's magic, the simulated immune system somehow targets the cancer, right? Because the immune checkpoint blockade is not targeting the cancer, um, which is kind of magical. And what's interesting is I think, historically, and this is maybe a little bit of a tangent, but I think if you were completely mechanistically inclined, right, just to be like, let's go specifically at tumor intrinsic mechanisms and all of those different things, then you never would have come across or been able to develop that kind of immunotherapy type paradigm. So the things outside of our current paradigm that may still have an influence and may be able to cause large changes in outcomes, I think still there's that possibility, and I hope there's that probability, right, in some of these maybe nanoparticles, maybe different ways of doing things, delivering of drugs, targeting of drugs, various other things. So I'm hopeful in that particular setting, outside maybe or using different approaches. Now, the other thing is, but to your point about the specific subgroups, how do we actually do this? And I think what I was trying to do when I was giving the examples, and I chose the examples to present today, was to say sort of, even though the heterogeneity feels overwhelming, and I took out a slide, which is already out of date, but it's basically a review saying like, here are all the different patient and tumor and microenvironmental features that have been associated with response resistance to immune checkpoint blockade, right? And it's a huge list, and it's already outdated, right? And the thing is that you know what I think is that the way that we can get a handle on these things is that to really try to, instead of just looking at responders, non-responders in this way, but how to think about it in a smart way, right? How to think about it like, well, we should really try to separate out the heterogeneity. Let's try to create more homogeneous patient populations, tumor populations that seem to start with the same biology in some sort of way. And then we can start to focus down and to look at what the differences between responders and non-responders, and again, improve that signal to noise type idea. Now, the hope is you can do that in a large scale way, but I think part of it is, and the problem is this is expensive and these things, but it's like you want to do that, deep characterization, multimodal characterization across as many patients as you can, across a number of different settings to basically understand and be able to dig into this.
Thank you very much. I think we have time for just one more question that just came in, and regarding the genes that had whole genome doubling,
Yep.
is it possible that the enhancers of the gene are being impacted? And has anyone examined what leads to the increase in the expression of certain genes?
So whole genome doubling is an entire area where there are a number of different ways that you get to whole genome doubling. I mean, you can think about it from an intuitive standpoint. Somewhere in mitosis, where the duplicate, the daughter DNA are supposed to separate, they don't separate out for a number of different mechanisms, and so you end up with a daughter cell that has double, right? And so in theory, that in and of itself does not lead to differences in expression, right, per se. But the idea is like the aberrations that you get as you go through that, and also as you continue to do mitoses and these now genome double tumors, which is very aberrant, right, and very strange, and lead to even more aneuploidy and other changes, then that leads to a range of different expression, if that makes sense. And actually, speaking of large scale chain things, actually one thing that I'm kind of excited about is that genome doubled tumors actually represent about 30% of solid tumors, depending on your study, but in one study, the pan cancer analysis, 30%. And if you think about the vulnerability in that setting, so it's dependent on a longer period of time in a particular phase of mitosis, and there's a particular kinesin that is dependent on, and so that is a potential vulnerability that is potentially targetable. And if that's 30% of solid tumors, that is potentially a very potent location, right, to target a fairly large set. So I think there's still creativity. I think there's still like these things as we're learning, as we're kind of going through. So I'm semi optimistic there.
All right. Thank you very much, Dr. Luke. That is all the time that we have for today. Any questions that we don't have time to address will be sent to the speaker after the symposium is concluded. And as of all other presentations during the event, I just want to offer a reminder that this talk will be available on the scientist website, and the link for the on-demand version will be available via email within a couple of days. So with that, let me introduce our next speaker today. Dr. Muad Barbiru is a research associate and circulating tumor cell core manager at the Sidney Kimmel Cancer Center, Thomas Jefferson University. With a strong background in medical oncology and population science, their research focuses on the role of liquid biopsy precocial circulating tumor cells in cancer progression, early detection, and treatment response. Dr. Barbiru has extensive experience in single cell analysis, liquid biopsy technologies, and translational cancer research, contributing to advancements in precision oncology. And they are actively involved in clinical and molecular studies, aiming to bridge the gap between research and patient care. So let's just make sure everything's up and running from a technical perspective. I do see your slides and I can see you on camera. Just want to make sure you can hear everything and do an audio check.
Hello. Yeah.
Wonderful. Please, the floor is yours.
Thank you. Thank you for the nice introduction, and thank you for having me as a speaker in your webinar. So I want to thank Dr. Dave for his wonderful presentation, and I'm gonna still talk in the same window, but from different aspect about cell heterogeneity and about cancer treatment and whole genome doubling. So today I'm gonna talk about circulating tumor cell as a molecular window into metastatic breast cancer evolution and using whole genome bisulfate sequencing technology. So as a background, in 2022, over 2.3 million new breast cancer cases were diagnosed and about 685,000 deaths were recorded in 2020, with around 38% rise in cases and 68% rise in death projected by 2015. In the US, about 311,000 invasive and 57 non-invasive, within 42,000 deaths were reported in 2024. Altogether, make breast cancer a major public health problem and attract researchers like us to find a better solution for diagnosis, for disease management and diagnosis. So breast cancer is diagnosed traditionally beginning with physical examination, followed by imaging techniques such as mammography, ultrasound, and MRI. While those methods help identify anatomical abnormalities, they do not provide comprehensive pathological insight into tumor characterization or treatment response. In addition to several tumor markers such as CA15-3 and CAA are used for monitoring breast cancer, but their specificity remain low with limitation in early detection and prognosis. Accordingly, imaging and blood tests assist in tracking disease progression, but do not offer sufficient molecular characterization of tumor. Currently, the gold standard for breast cancer diagnosis and treatment evaluation is tissue biopsy, followed by histopathological analysis. However, this method has limitations, including invasiveness, potential sampling error, and its inability to capture tumor heterogeneity dynamically. So, despite advances in breast cancer management, challenges remain in achieving early detection, precision molecular characterization, and personalized therapy, which are critical for improving patient outcome and survival rates. On the other hand, tumor classification is switching from anatomical to molecular classification, which promise a new translational strategy in clinical research. that instead the need of breast cancer early detection and therapy management improvement using fast and repetitive methods based on biological material that reflect the real-time tumor. According to the urgent clinical necessity, the intention of researchers has turned to the investigation of liquid biopsy as better solution for breast cancer characterization. So what's liquid biopsy? Liquid biopsy is based on body fluid, mostly blood, but also other fluids such as saliva and urine for biological testing. Among compound released from the primary tumor into body fluid and mainly in the peripheral blood. CTC present a promising source of cancer biomarkers that are detectable in the blood and carry a large diversity of biological molecules that can be used for the diagnostic and monitoring of multiple invasive cancers. In this context, we aim to perform single-cell-based resolution sequencing of circulating tumor cells to map whole genome patterns. And we also aim to assist intra and inter-patient variability in circulating tumor cells methylation profiles and to compare the finding with the copy number alteration data to understand the tumor evolution and diversity. As the overall objective of the study is to identify potential biomarker for circulating tumor cells methylation patterns that could be used for breast cancer prognosis and treatment response prediction. To do that, we ran 4 milliliters of blood in the Genesis system with cell select slide enable the isolation of circulating tumor cells from all kinds of liquid biopsies, such as blood in this case. Cell select slides from BioRed utilize about 56,000 individual microchamber to isolate cells from whole blood based on their size that superior to 8 micrometers. The cell select slides for size capture cells in the range of 8 to 13 micrometers. In that case, red blood cells and smaller white blood cells flow through the process to the waste, while cells larger than 8 micron are isolated, such as the size of circulating tumor cells and circulating tumor cell clusters, and for the majority of solid cancer types. CTC is above 8 micron and captured by the cell select slides. For this representative metastatic breast cancer patient, we enriched and enumerated about 195 circulating tumor cells. For the CTC, circulating tumor cells, the cell size was set at 20 nanometers at the imaging software. Then selected CTC are positive for DAPI, for nucleosis marker, positive for GFP, for C2P, for PNCK markers, that's the specific marker for tumor cells, and negative for CD45, which is the marker for leukocytes and white blood cells. Regarding white blood cells, the selected size was under 20 nanometers, the diameters. Then the selected cells are positive for DAPI, nucleosis marker, and negative for GFP, and positive for CD45, which is marker, leukocyte markers or white blood cells marker. In addition to the regular CTC marker in our breast cancer enumeration, we were able to successfully add 3 additional markers for CTC selection criteria, such as vimentin, and ER and HER2. So as shown in this figure, CTC still remain positive for DAPI, positive for PNCK, and could be positive or negative for vimentin, because there is two kind of circulating tumor cells, or two kinds of cells. There is one are positive for vimentin, which are mesenchymal migration, and one are negative for vimentin, which could be positive for EPCAM, for non-mesenchymal migration. And cells also could be AR positive or AR negative, and HER2 positive or HER2 negative. And cells can co-express any of those markers. Any CTC can co-express any of those three markers, or could be negative for those markers or positive for those markers. And for the white blood cells, it has to be always negative for cytokeratin, PNCK, the GFP expression, and could be positive or negative for vimentin, and negative for AR and HER2 expression. After we CTC enrichment for the sample that reached the minimum threshold, we processed the single cell isolation using deep array system. So circulating tumor cells isolated from peripheral blood are isolated from peripheral blood, enriched using the same slides. We flow through the cells to the collection tube using the Genesis platform from Biored, because there is slides that gonna be, the blood is splitted into one slide gonna be used only for enumeration, and the remaining amount of the blood, the other 4 milliliters of the blood, gonna be used for enrichment later using the same workflow. But at this time we don't fix the slide, we just flow back the slides in the collection tube, and then we do outside staining of the slides of the collected cells using the same antibody that we used for enrichment. So we used circulating tumor cells isolated for peripheral blood. So here we're used using deep array system. So what we do, the enriched cells will be loaded in the specific cartridge. Then the cells are loaded in the dedicated cartridge and visualized by fluorescence microscope. Although a small amount of those cells might share markers of both lines, these methods also allow the size evaluation of the CTC in bright field. So the CTC are moved into a parking area using dielectric forces and recovered as single or clumped cells. Further down, the downstairs analysis can be assessed on those cells, such as NGS, including both DNA and RNA-seq, or Sanger sequencing, and digital PCR. So basically the cells, we end up, according to the fluorescence, we pick the cells in subgroups, then we move them to the parking. Then we end up by a single cell in a single PCR tube. So according to the collection or to the study design of the project, you choose to collect multiple cells in one single tube according to their expression of the biomarker expression of the surface marker, or you can end up by a single cell in a single tube, which is the case of this project. So this is a representative image from deep array cells analyzer software. Typically double positive CTC. If you look at here, the CTC is positive for HER2 and positive for AR, positive for nucleosis, and positive for PNCK. And this CTC is double negative. It's positive for PNCK, cytokeratin, and negative for nucleosis, negative for CD45, and negative for both HER2 and ER. However, this is the white blood cells, which is negative for PNCK, positive for DAPI, and positive for CD45, and negative for for the other breast cancer markers used in this study, which are HER2 and ER. And we also check the circularity of the cells. We need to collect an intact cell, not cluster of cells, and good circularity and good shape of the cells. After that, we do whole genome bisulfate sequencing of the picket cells. So basically, we end up by a single cell in the tube. So we move it we move it on with those cells to bisulfate treatment. We use it enzymatic treatment to treat the cells, to do bisulfate conversion of the single cells. Then we do whole genome amplification of the single cells, and we move it with the whole genome library construction. Then we do after library construction, we do library QC to see like the peaks of the peaks of the libraries, if we have a clean library to be sequenced, or if we have cells in the tube, or if we didn't move the cells. So a library that didn't pass the QC was eliminated, and we end up by only the cells that they have a clean and a clear library. So our study, it showed for the first time that we demonstrated that whole genome duplication can occur independently in a separate sublineage within patient-derived samples of metastatic breast cancer CTC. So the key finding of our study, in a representative patient, both 2N and 4N diploidy CTC were found in distinct subclone, indicated heterogeneity. And when analyzed at uniform 2N ploidy and 4N cell appeared depressed, suggesting the whole genome duplication occurred later in tumor evolution, not at the earliest stage. We also notified two independent whole genome duplication event were interfere. So in this figure, one subline lost chromosome X, followed by a whole genome duplication in only 8 cells of the patient, of that patient. And in this figure, there were a gain of chromosome 7 prior to separate whole genome duplication event, and that's occurred in the 11 cells from the same sample and coming from the same patient. So then we pass to the mathematical modeling of those cells events. So in in these figures, we developed hybrid linear and hybrid polynomial model to capture both punctuated and gradual copy number aberration evolution. This model demonstrated the high accuracy of the copy number alteration evolution, that superior to 0.95 in all tested breast cancer patients. So this is made the conclusion that metastatic breast tumor exhibit gradual copy number aberration evolutional following early punctuated events, challenging the previous beliefs of whole genome duplication primarily occur early in tumor development. So we also examined the methylation clusters and the UMAP clustering of all cells based on their DMRs, which identified 11 distinct clusters. Each cluster largely correspond one-to-one with the patient of range. We then picked the top 60 markers region for all clusters in the heat map within representative markers. So cluster one with low methylation cluster one with low methylation of the ADM-TS7 region, hypomethylation markers, and cluster 9 with high methylation at the ZNF302 region, hypermethylation markers. So the methylation cluster closely reflect the patient of origin, which distinct hypo from hypermethylation markers identified for specific patients. By doing this study, that we have concluded that whole genome methylation profile at a single cell resolution revealed distinct methylation clusters that strongly correlate with patient of origin, demonstrated the feasibility of high resolution methylation mapping in circulating tumor cells. We also concluded that the identification of intra and inter patient heterogeneity in methylation patterns, alongst independently with whole genome duplication events and distinct copy number alteration profiles, highlighted the complex and the diversity of evolution pathway in metastatic breast cancer circulating tumor cells. And we also conclude that patient-specific methylation markers such as hypomethylation of ADMTS7 and hypermethylation of ZNF302 suggest the potential of developing clinical relevant biomarker for prognostic and personalized treatment strategy. So by doing this study, we showed that there is we confirmed, we don't show, we confirmed that the heterogeneity of, there is heterogeneity of the tumor, and that's why sometimes the patient showed progression over time, or the patient didn't reply to the treatment, and those cells, the heterogeneity of those cells can be detected in the blood, and if those cells that are able to skip the treatment and they go into circulation, they might be responsible for the metastatic and for the metastatic later on. For those patients. So by doing this blood test based on circulating tumor cells, we can detect the heterogeneity, and we can probably cover as much as we can of molecular profiles of the tumor, of the patient tumor. Thank you.
Thank you very much, Dr. Babidou and Biorad, for that presentation. So let's just quickly again jump into our Q&A session, and to offer a quick reminder to our audience that to submit any questions for our speakers, you can do this using the Q&A tab on the right side of your screen. I just wanted to ask a question about the DEP array. I guess the first question is, what is the throughput of the instrument? How many single cells can you isolate per experimental run, or in a single workflow?
So DeepArray has two different workflow. It's one with work with live cells and one work with fixed cells. In our case, in our CTC pipeline, we use the workflow that work with fixed cells, because we fix our cells before we enriched them with the antibody and all of that. So we are able to pick as much as 96 CTCs per sample. 96 single cell per sample, because we pick both groups, CTCs and white blood cells, as a control from the same patient.
Thank you very much. So my second question is regarding, you mentioned that you may pick up cell clumps in the single cell isolation process. I was wondering, how large are those clumps, and is there a size threshold in terms of what the instrument will look for when trying to isolate single cells or entities that it thinks are single cells?
Actually, this is for Genesis. That's not on DeepArray. DeepArray, it's a single cell picker.
Mhm.
DPERI is the microfluidic pick. It's the microfluidic picker. It's just a single cell picker. You cannot go higher than one single cell in the tube, or you can do multiple cells in the tube. But Genesis, the enumeration, the capture platform, the enumeration platform and enrichment platform,
Mhm.
it holds all the cells that are bigger than 8 micron. Anything is bigger than 8 micron that will pass through the slides, it's gonna be captured by the cell. However, I saw clusters of 12 cells, like about 38 to 40 micrometers that was captured by the microchamber. If you go too big, you're gonna block the flow of the
Mhm.
sample and you cannot finish your run. So we usually we try to don't have clog, or we don't have a big cells on our samples. So that's why we don't block the microchamber, or we don't block the flow of the blood into the microchamber of the slide. But I saw about 8 CTCs, cluster of 8 CTCs very clearly in Genesis platform.
All right. Thank you very much for that answer, Dr. Barbidou, and for your presentation, and for being with us today.
Thank
And,
you.
and again, a reminder to our audience that first, that any questions that we don't have time to get to will be sent to the speakers after the symposium, or you can contact them directly through the email shown on the screen. Another reminder as well that this presentation will be available on the scientist's website, and we will email all registrants the link for the on demand version within a couple of days. At this point, we'd like to take a few minutes just to have a break or maybe a step away, uh, have a coffee break, and we'll be back soon with our next talk, which is scheduled for 12:20 p.m. Eastern Time. So by my watch, that is 4 minutes away. We'll see you then. All right, everyone. We are now ready to resume our event with our next speaker. Omid Visse is a professor of bioengineering and chemical and molecular engineering at Rice University and a Cancer Prevention and Research Institute of Texas scholar in cancer research. He's also the director of Rice University's Biotech Launchpad, a new initiative with a mission to accelerate the translation of Rice University discoveries and technologies into clinical practice to provide rapid patient access to leading edge therapeutic products. Throughout his career, he has authored or co authored more than 80 peer reviewed publications, and he is an inventor on more than 50 pending or awarded patents. He's also a serial entrepreneur who has co founded multiple biotechnology companies. Visse has been elected as a Fellow of the Controlled Release Society and a member of the National Academy of Inventors. He leads an interdisciplinary translational research program to engineer and commercialize next generation cell based therapeutics for various human diseases. His team leverages the latest techniques in synthetic biology, immunoengineering, and material science to develop innovative cell based platforms for real time and feedback regulated production of biologics. Let's just make sure everything's up and running from a technical standpoint. I can see your slides, so let's just make sure you can hear my voice, Dr. Visse.
Yep, I can hear you. Can you hear me?
I can hear you loud and clear, so the floor is yours. Please take it away.
Great. Well, thank you for this opportunity. It's a pleasure to be part of this panel for this webinar series. And I'm going to highlight some of our work in developing cell-based therapeutics with a particular focus on oncology. Um, a big focus of my lab is to think about bringing biologic manufacturing into the patient. We know that there's a lot of drugs that are having great impact on patients' lives, a lot of biologic drugs, and biologics have this complication where they have to be either infused or injected. And for chronic use, even in the case of oncology, for example, many patients now will take checkpoint therapy for up to 18 to 24 months. This can become quite a huge burden, both in terms of patient compliance, but also in terms of cost. And so we've been thinking about innovative ways to really change that paradigm and bring that manufacturing into the patient. If we think about the current biologic production pipeline, where biologics are made in steel bioreactors, they, will start with engineering a cell, and then a great deal of work goes into purifying, stabilizing, creating a fill-finished version of the product that then has to be injected and infused. In some cases, it could be multiple times a day that's being administered to the patient. And there are some formulations that are now once every few weeks. But when you're thinking about a disease where you may have to take this for many, many months to years, this can add up. And we all know the pharmaceutical industry has a certain amount of greed, but even the cost of goods for, let's say, an antibody-based therapy could be as high as $1,500 a dose for maybe 1 year of product that a patient would use. We're thinking about innovative ways where we can really reduce that by creating the cells, combining them with the biomaterial, and then administering that to the patient. And I'll go through some examples of where we've done this and actually advanced them to human testing already. And so this idea of a living drug factory is intriguing to us because it allows us to not only think about the long-term delivery aspect, but also in certain cases where localized delivery to a particular cavity or to a particular region of the body constantly can be beneficial, such as the case for immuno-oncology, where delivery adjacent to the tumor draining lymph nodes could boost really therapeutic outcomes and release and reduce some of the side effects. And then the other unique advantage is we can hack into cells' ability to sense and perceive their environment to create feedback regulated systems, such as, for blood glucose control, where insulin could be produced in response to hyperglycemia events. or on-demand production, where we can build in the system an ability to turn on production when needed. So key to making this possible is we've for years been working on innovating biomaterials formulations that allow immunoisolation of these transplanted cells from the host immune system. These are porous hydrogels where the pores are too small for immune cells to infiltrate into. And they have these small molecule modifications on the hydrogel backbone that actively work to suppress foreign body responses to the material, allowing these encapsulated cells to persist for months to years, or a predictable amount of time, depending on which small molecule formulation we use. And to find these small molecules, we actually applied a discovery-driven process where we would create a large library of small molecule modified hydrogels. And the hydrogel we like to use for this application is alginate as the backbone. And this is because alginate can be created in a hydrogel format in a very cell-friendly manner. It cross-links with divalent cations, such as calcium and barium, and very widely used both in the medical industry in terms of implantation, but also in the food industry. Many people that may be interested in molecular gastronomy, alginate hydrogels are used to make things like vegan caviar, or in terms of sausage spices, they can be encapsulated in this hydrogel and administered. So there's a long history of their use with humans. But if you were to implant unmodified alginates, it creates a foreign body response. So we created this process by why we develop a library of these, and then develop formulations of hydrogels, implant them into animals, and the position would be a barcode for us where we could decouple exactly inflammation and foreign body responses at a particular site, and essentially develop a quantifiable way to explain how certain modifications to the alginate would either reduce host immune reactions and create formulations that are ultra long lasting, or boost the localized inflammation and create formulations which activate the immune system in more predictable ways. And both are really useful and exciting to us for different purposes. In the case of the low inflammatory and low fibrotic, those allow for, ability to produce biologics in the body for many months to years. And in the cases of the inflammatory formulations, they're particularly interesting, and we've used them in the context of cancer immunotherapy, where we want to boost a localized immune response. So how this works is, if we were to implant these materials like we've done here, you can see that these are alginate spheres. This is a pro-inflammatory variety where the immune system has recognized this as foreign. You can see in this particular video, these are macrophage cells that all express green fluorescent protein. And this is a live video, 7 days post-implantation, about 6 hours of imaging that's in a loop of a 30-second video. But you can see that there's quite a lot of activity around the implants. A lot of macrophages are trafficking to the intraperitoneal site where they're implanted. And in many cases, that macrophages have adhered to the surface. They've become activated. They secrete additional cytokines, which further recruit more immune cells. And we contrast that with our ultra low inflammatory alginates, which allow for a long-term viability of the encapsulated cells. And in this case, you can see that there's a healthy level of macrophages there. So it's not like it's toxic to the immune cells, but the material actually is such that the immune cells can't adhere to. And when they don't adhere, they don't become activated. And this material can just now reside, and the encapsulated cells would be able to persist. And this is how these different formulations work. And it allows us to do things such as load them with cells that produce biologics and we have engineered a cell chassis in my lab, these ARPE19 cells, which can be genetically manipulated to produce a range of biologics. And actually, we have a project in the lab where there's currently like 330 FDA-approved biologics that are used as treatments today. And we've actually gone through the process of made ARPE19 cells, which produce every one of these biologics. And this allows us to have a large repertoire of biologics that we can deliver for a wide range of diseases. And so any antibody, cytokine, chemokine, hormone that is produced today through recombinant DNA technology, we can create ARPE19 cells that make them. And we've spent a lot of time optimizing to make sure they're super potent. And this particular cell line is unique in that it can then be transplanted into patients because it's non-tumorigenic, and it has a really unique feature, which is contact inhibition. So as we develop clones that make these different biologics, we can expand them infinitely to billions and trillions of cells, and they'll continue dividing. But when you encapsulate them and you put them on hydrogel, they then stop dividing because they have contact inhibition. And that's really critical because it allows us to control dosing. So essentially the dose that we administer at day 1, that is the max dose it can be, because within our hydrogel devices, they no longer can divide in the patient. And then the other thing is there's a history of having these cells advance to clinical trials. Multiple companies have advanced the cell chassis. In fact, Neurotech just announced that their product for MAC type 2 just got approved by the FDA. So it is now a cell chassis that's used in an FDA-approved cell therapy product. This is a product for the eye where they are also encapsulating these cells, and they produce the biologic that treats MAC type 2. It's administered directly into the eye, and they've shown that this cell line actually can persist for up to 13 years in human patients, producing the biologic. Now the eye is immunosuppressed, is an immunoprivileged site, meaning that there is no foreign body response to it in the eye. And that's where we've really advanced the use of these, where we can now implant them to various parts of the body to achieve similar goals. And using the latest advances in synthetic biology, we've created ultra high potent versions of these cells, which really allow us to reduce the implantation volume. And so a very small number of cells in these capsules could be very efficacious in terms of producing biologics. Here's an example of a project we have where we're delivering antibodies. These are broadly neutralizing antibodies that are targeted against HIV. And there are clinical versions of these that are now being tested. And we've created the same exact biologic, but generated through the ARPE19 cells. And they're equivalent in terms of potency and the ability to neutralize the HIV. And by really spending time to create ultra high potent cells, what this translates to is for a 1-year treatment, we can create formulations that we administer once subcutaneously. These enable the living cells to persist in the patient for the 1-year period, producing a daily amount of roughly 400 micrograms per day of the biologic for a 70 kilogram patient. And that what that allows us to achieve is once yearly formulations of biologic therapies at a significantly lower price point. Cost of goods for our approach is roughly around $50 by just making the cells once and then having them live in the patient. And then those living cells are then producing a daily amount. This really changes the paradigm. And we've shown that we can do this in fully immunocompetent animals, and we have validated that we can test the efficacy of this over time and show that this is actually able to be both efficacious and therapeutically relevant titers for up to 1 year when we use the right formulation. Now, going back to our library of small molecule modified allogeneics, we also got intrigued about these high inflammatory formulations because they could synergize with biologics for immunotherapy and provide more efficacious results in terms of immunotherapy. One class of molecules which is particularly interesting for immunotherapy is cytokines. When we think about the major pillars of immunotherapy, to have a successful immunotherapy outcome, there's really three key things. You need to have antigens, and in tumors which actually have a high number of mutations, should be sufficient antigens for the immune system to recognize these cells as different from healthy cells. You need to have checkpoint blockade, which is a way that the immune system prevents attacks from persisting against host cells. And that's been largely solved through the development of checkpoint therapies, antibodies like Keytruda and ipilimumab that go after those checkpoints. The third pillar, which has been really tough to manage, is cytokines, because you also need a cytokine to activate and facilitate differentiation of the immune system. And that in the body, cytokines have evolved to be highly potent, fast-acting, short-lived. And there's a spatial and temporal regulation over their concentrations. But most of our experience with cytokine therapies has been through systemic administration. And molecules like IL-2 and IL-12 and IL-15, when you administer them through the IV route systemically, you have large reservoirs of immune cells that you activate all at once. And this creates a significant amount of toxicity and triggering what is called cytokine release syndrome, which can lead to a lot of complications. And actually, where you want the effect to be, which is in the tumor draining lymph nodes, probably you're not getting very high concentrations of cytokine there. Instead, you're increasing it elsewhere. So this really led us to think about, can we use our platform of cell-generated biologic factories to create concentration gradients adjacent to tumors where there's tumor-draining lymph nodes present and there's high concentrations of antigens, to create that local concentration of high cytokine levels where they're actually needed and keep them out of the systemic circulation where they can cause toxicity. And the cancer that we prioritize for first is ovarian cancer, largely because of the significant unmet need. Unfortunately, ovarian cancer patients today face a dismal prognosis where there's about a 50% survival outcome if it's stage 3 or higher. And in cases where there is treatment through carboplatin and resistance develops, unfortunately, these patients are rarely curable. And so they face a very dismal prognosis. We were inspired by trials that were done by Edwards and David Miner at University of Pittsburgh. And what they had done was actually taken those refractory recurrent patients. These were the incurable patients that had a progression-free survival outcome of roughly 4 months. And they enrolled them in a trial where they administered IL-2, which is FDA approved as the drug Proleukin, through the intraperitoneal route. They hooked up these patients to a pump system where they were infusing in 16 weekly infusions of IL-2. And unfortunately, because these cytokines are so unstable, they have to be very carefully formulated. And the formulation was such that it had to be a 2-liter administration. So 2 liters a day was administered for over 16 weeks. And in this trial, which was really hard to pull off because many of the patients had complications from the administration, catheter obstructions, and other things, there was really remarkable results observed in that 6 out of the 24 patients had objective responses, 4 complete responses. And I believe the 4 patients that had complete responses actually never had a recurrence and they were cured. These patients, I believe, are still alive today, and many of them were really advanced patients which had stage 4. And so this showed that this concept of local delivery to the peritoneal cavity, where you have the ovarian cancer cells and the tumor draining lymph nodes are able to see those antigens, could work if you could deliver the right concentration for the right duration without all the complications that was associated with the pump system. So this became our motivation, is to really build a system that can accomplish this. And so we created these cytokine factories where we took our engineered ARPE19 cells, programmed them to produce proleukin and other cytokines, and showed that indeed, if we were to implant these, and these are 1.5 millimeter spheres that could be administered through either laparoscopic surgery or IR guided administration through the peritoneal space. These beads can then live inside that peritoneal space and produce a particular dose of this biologic IL-2. And as they produce it, because the production rate is higher than and the elimination rate is so fast in the blood, we're able to create these local concentration gradients where within the peritoneal space, we can create 100-fold higher concentrations than exist in blood, achieving levels that were consistent with what Edwards was administering, but that 2-liter injection volume could now be shrinked to a 5 cc volume of these beads instead. And when we administer these beads, and these were put into our inflammatory alginates, because we wanted to also provoke an immune response. And we know that these capsules do get fibrosed, but that's okay because we want them to be transiently active. In this case, they're designed to shut off within 3 to 4 weeks after administration. And that allows us to create a concentration gradient where one administration creates high concentrations of IL-2 into the interperitoneal space for roughly a month. In the blood, it's 100x lower at all time points. And you contrast that with how IL-2 is administered in the clinic today, which is 3 times daily IV infusions. So this becomes a lot more convenient and easier to administer. And we can control dosing just like any other biologic. Here we control it based on the number of spheres we administer, because each capsule has a certain production capacity. And based on the number of capsules, we can change that dosing paradigm in the patient. And after the 30-day, if we wanted to administer another dose, we can, and we get the same exact kinetic. So 30 days later, we can come back in and put another dose of the capsules, and we get the same kinetic all over again. And then the other interesting thing that we discovered in going about this approach was that while recombinant biologic manufacturing has the challenges where you have to produce the biologic and figure out how to stabilize it from the time that it's produced till it goes back to the patient, this often requires re-engineering of the biologic, as has been the case in proleukin, where the native form of IL-2, which human genome is encoded, actually has a disulfide bond. And they noticed poor stability through recombinant production. And instead, they had to re-engineer it, change the amino acid sequence to a format that could be both produced and was stable enough against natural degradation. What this created was a xenoprotein to the human body, because this is not a protein that the body itself encodes, and resulted in anti-drug antibodies being detected in the patients treated with. And this is actually from the FDA label, where 74% of the patients actually developed antibodies to the therapy. So it was probably mostly short-lived in terms of function. Within a few weeks, likely re-administration resulted in neutralization of the drug. And the other nice thing about the strategy of having the actual natural biologic is that it interfaces with the receptor for IL-2 in a more complete manner, which results in higher potency. we actually noticed a 3x higher potency of the cell-generated IL-2 compared to the recombinant version of this. Once we had this system developed and we showed that we could create the right PK and PD activities, we then tested it in various models of cancer, starting with ovarian cancer mouse models. This is a syngeneic IDA tumor model, which is resistant to immunotherapy, as is the human disease. And we wanted to test to see if our IL-2 generating capsules could have an impact on these mouse tumors. And we noticed that, through a single administration, we were able to eliminate the tumor in these cohorts of mice in ways that constant recombinant administration of IL-2 could not achieve, because when you inject it, it quickly degrades and disappears. And unless you can do what Edwards did, which is a constant infusion pump, which we couldn't really set up for the mice, then you don't get the same efficacy that you would through the constant production in the local compartment of the IL-2. We then evaluated the immune populations, and we noticed that the immune profile is as we would expect: expansion of effector immune cells, both CD8 and CD4 positive cells. Another interesting thing that we noticed is while the therapy was being administered locally, we noticed that the immune cells that were being generated in the local compartment could then traffic through the body. And in fact, we saw effector T cell expansion into this spleen, which suggests that while the cells are generated locally, they're able to now migrate throughout the body and find the cancer where else it may be. We then tested it in a different model. This is a colorectal cancer model, MC38. And this is a particular tumor model which can grow in multiple compartments of the body. It is, of course, generated from colon, but then can metastasize to the IP space. And in an animal setting, you can implant it in various compartments as well. We wanted to test whether local treatment could lead to systemic immunity with tumors being in other cavities. And so we did a study where we first treated these tumor-bearing mice and showed really good efficacy in more than 80% of the mice being completely cured. We then took those cured mice and gave them the tumor again in a different cavity, the subcutaneous space. And what we noticed is that in the mice that had already been treated successfully once, they were now resistant to the tumor again, versus the naive mice, the tumors could grow fine. And that's because in the process of eliminating the first tumor, the immune system had built memory cells, which were then able to recognize the cancer upon second administration. Now, a lot of the work that I just showed was in the IP space, but we noticed that actually there's been, in addition to the work that Edwards had done with ovarian cancer, there was also evidence that local administration constantly of IL-2 to other cavities, such as the case with pleural malignant mesothelioma, could also result in efficacy in terms of having these patients do much better. They didn't get quite the same cures that Edwards did, but in many of these cases, they saw partial responses. In one case, they saw a complete response. And mesothelioma is a particularly devastating disease, which rarely is cured. So to have that level of efficacy is quite striking. And so we wanted to test whether we would get the same concentration gradient differential in other cavities as well. And so we administered these capsules, which are dyed black here, so you can be easily visualized, into the pleural cavity. And this is kind of how they rest, just sitting outside the lung in this pleural cavity. There's the pleural cavity wall, and this is the lung. This is where the tumors usually grow. And we noticed that, in this cavity as well, we saw 100x differential in concentrations locally versus the blood, creating the opportunity to deliver this for this disease as well. And we then tested malignant mesothelioma mouse models, AB1 tumors, with this treatment. And in particular, checkpoint therapy today is how these patients are managed. And we tested it in the context of with checkpoint therapy. notice that we get really nice responses and 100% of these tumors being eliminated when we administer our treatment combined with the checkpoint therapy. And again, demonstrated that these mice that have been successfully able to eliminate the tumor are now protected against a second tumor administration in ways that the naive mice are not. When we looked at the immune profile, we noticed expansion of the T cells, but in particular honing in on the innate immune populations, we saw some really interesting results in that, in addition to activating properly the adaptive immune cells, the T cells and the dendritic cells, we also saw activation of inflammatory macrophages. In particular, mesothelioma tumors have a phenotype where they have high concentrations of suppressive M2 macrophages within the tumor microenvironment, within the pleural cavity. And that was recapitulated in these mouse models. But through our therapy, we noticed that our capsules, because of their inflammatory nature, were actually shifting and expanding M1-like inflammatory macrophages, which create a more favorable environment for effective immunotherapy, which could not be achieved through just IL-2 administration by itself. So, having shown that this can work in multiple mouse models, we wanted to assess whether indeed we had moved the needle in terms of reducing the toxicity profile, and also whether the dosing that we had established in mice would translate to larger animals. So we did studies in non-human primates where we would administer, again, different doses of the capsules, and we can control dosing based on the number of capsules. We can also report it based on, microgram production capacity per day. And we saw actually a really good correlation in vivo in terms of measured drug levels based on what we would predict a 0.94 correlation in terms of being able to create these local IP concentrations and systemically very low levels. And then we saw expansion of the effector T cells in the IP space and no expansion of Tregs, and that this was, tolerated really well and was safe in these non-human primates. So armed with this data, we wanted to test whether this could work in human patients. And the patient population we went into was the recurrent refractory ovarian cancer patients. Did a trial. This was a dose escalation trial where we had 4 dose intervals. At each dose intervals, we were doubling the dose. And this is what the data look like in the human patients. 14 patients have been dosed to date. And we were excited to see, again, this dose-dependent increase in the IP space that was 100x higher than the concentrations in the blood, and expansion of the right immune cells in terms of CD8 positive effector cells. Those cells are producing interferon gamma, expansion of NK cells, and no Treg expansion. And looking at the therapy outcomes, we were really excited to see that as we started increasing the dosing and we got to dose level 2, patients were starting to actually have delayed progression. And we had one patient that went out to 24 weeks of stable disease without any progression. We had one patient that started to have a partial response, recapitulating some of the data that Edwards had seen, but our numbers aren't quite high enough to the 25 he's dosed. So far, we're at 14 patients. But this is starting to look really encouraging that at higher levels, we're seeing the same level of efficacy that he did with our new system, which is much more convenient and easier to administer to the patients. And we also see a dose-dependent decrease in CA25, which is a biomarker of ovarian cancer in these human patients. And profiling the immune cells of these patients that have received therapy, we notice a dose-dependent increase of CTLA4 in CD4 and CD8 positive cells, suggesting that synergizing with checkpoint therapy, in particular ipilimumab, which is targeted towards CTLA4, makes a lot of sense. And that's what the next version of the trial is going to be. Higher dosing, combination therapy, multiple administrations, hopefully achieving much better therapeutic outcomes than what we're seeing today. But already the data looks really encouraging. So the other thing that we're thinking about, and I'll just spend a quick couple of minutes on this, is we notice how important dosing is in this system. And because we're using a living therapy, we really do have a unique ability to develop systems that could then be either on-demand triggered for biologic production or built-in sensors that could actually regulate dosing on a patient-by-patient basis. So we imagine a future where we are combining these cell therapies with bioelectronic devices that provide support and regulation for these cells. And actually, a couple of years ago, 18 months ago to be exact, we received one of the first awarded ARPA-H grants, which is to develop this exact solution, which is a feedback regulated bioelectronic device with living cells where we can actuate concentration of immunomodulators, being able to do combination therapies like IL-2 and IPI together generated from a device, and at the same time have onboard sensors that allow us to track concentrations of the drug we're producing, as well as biomarkers of safety and efficacy. This project is going well. We're about 1 year away from entering clinical trial with this next version of our device. And we're able to show some of these unique abilities, such as being able to control dosing by engineering circuits into the cells so that we can use electrogenetic signals to turn biologic production on and regulate the dosing. So these are some of the early data from this effort. But again, the Fully device is being integrated, and we're excited to move that into human patients as well. So with that, I wanted to thank my team. One of my former PhD students, Amanda Nash, really led this effort of developing these cytokine factories. It's really exciting to see when you have really motivated students. We actually went from an idea on the shelf, which was to create these factories, to having them advance to human testing by the time she graduated. And so that was really unique to see, as well as other team members that contributed to this effort, and of course our funding sources. And with that, I'm happy to take your questions.
Thank you very much, Dr. Vise, for that presentation. So let's get to the Q&A session. I just wanted to ask, what are within the context of these the cytokine factories and the cellular factories in general, what are the primary differences that you would expect or that you were anticipating between working with rodent models and working with humans or larger primate models?
Yeah, that's a great question. So some things that translated really well were actually the PK levels going from all the different models. And surprisingly, that was really well conserved. The things that are, I think, variable are really the pharmacodynamics. And that seems to be both heterogeneous, even within animals, but also within the human population. And that's where we're really excited about these feedback regulated systems, such that we don't really dose to a set PK, but we have the ability to adjust dosing on a per patient basis to really account for the heterogeneity that exists even within the human population. Does that make sense? Hello?
Yes, sorry about that. Yes. With regard to these cells as well, how much do they integrate within the physiology of the body? I know that you've mentioned that they don't replicate, they don't proliferate. Do they migrate, and do they respond to internal signals?
So they don't migrate. They're actually within the hydrogel or within the device, but they do have bidirectional communication with the host, meaning any soluble factors are able to diffuse into the device, as well as any secreted factors generated by the cells are able to secrete out of the device. But physically, they are prevented from contacting host immune cells, which allows us to use allogeneic cells that don't get recognized as foreign. So the T cells that would normally recognize an allogeneic cell as foreign is physically unable to contact our cells, and because of that, they're able to persist for long periods of time.
Have you observed any shifts in function or functionality, efficacy over time because of these interactions?
Um, have we noticed any shifts over time in terms of the potency of the cells?
Potency, or even level of activity, either in either direction. Because typically
No,
immune related cells are very plastic in that regard.
right. Um, so we have not actually, in terms of the stability of the cells in vitro, they seem to be really good, stable, really highly stable, um, low genetic drift. Of course, in vivo, there is some heterogeneity in terms of, um, potentially if there's foreign body responses and things like that. But in general, I would say we've been really impressed with how consistent they have been. And that's largely because we go through this process of identifying a particular clone, which is robust and able to maintain concentrations really well. Um, but then, in terms of the human response to the factors, now there is some heterogeneity there, which is consistent with, the variability in terms of the treatment outcomes that people notice.
Thank you very much. We have a question asking, to what extent does the cytokine profile of a malignancy affect the efficacy of immune memory responses in episodes of relapse? And I'm wondering if that also kind of ties into how potential therapeutics with the cytokine factories, regarding whether or not you would have to change the regimen for a relapse as opposed to for kind of the initial oncogenesis.
Um, could you say that again? Sorry, not following exactly.
Sorry. Uh, so the question is, what extent does the cytokine profile of a malignancy affect the efficacy of immune memory
Mhm.
responses against potential relapse?
I see. Yeah, no, that's a great question. So, what we have noticed is, you know, these patients, when they go through chemotherapy, it does seem like their cytokine profiles within the tumors become much different. Actually, we've noticed in cases where prior to initiating any therapy, they have a particular cytokine profile. Then they go through chemotherapy. A lot of the effector immune cells are depleted. The tumors tend to become a lot more suppressive by nature, making it harder for immunotherapy to be successful. We're hoping to recapitulate uh, the local environment to a level of cytokines which are much more favorable for immunotherapy outcomes. And we think that that's part of the reason why we are seeing this great efficacy in our models, because in general, it is the case where as the tumor progresses, they tend to become more suppressive. And sometimes, like in the case of ovarian cancer, we're noticing that the carboplatinum treatment in itself is perhaps shifting the immune profile of the tumors and making it more immunosuppressive as well. So these are things that need to be um, addressed, and we think that's what is unique about our platform is the ability to do that.
Thank you very much, Dr. Visse. And just to wrap up this part of the event, any questions that we don't have time to get to will be sent to the speaker after the symposium. And as a reminder, This presentation will be available for on-demand viewing on the scientist's website, and you'll receive the link and additional information for that within a couple of days. So with that, let's move forward with the final presentation of today. And I'm pleased to welcome our final speaker, Dr. Elaine Mardis, who is a co-executive director of the Steve and Cindy Rasmussen Institute for Genomic Medicine at Nationwide Children's Hospital. She holds the Rasmussen Nationwide Foundation Endowed Chair in Genomic Medicine and is a professor of pediatrics at The Ohio State University College of Medicine. Mardis is an internationally recognized expert in cancer genomics and immunogenomics with over 550 published manuscripts, and is the former president of the American Association for Cancer Research. In 2019, she was elected a Fellow of the American Association for Cancer Research and a member of the U.S. National Academy of Medicine. She has co-led the effort to sequence and analyze the first cancer genome using massively parallel sequencing technology, and was integral to large-scale cancer genomics discovery projects, including the Cancer Genome Atlas and the Pediatric Cancer Genome Project. Her ongoing research interests lie in the integrated characterization of cancer genomes, defining somatic and germline genomic interactions, RNA characterization, and immune microenvironments that lead to cancer onset and progression, with a focus on pediatric cancers and precision oncology. Okay. Dr. Mardis, I can see your slides, I can see your webcam, and I think I can hear your voice.
Hopefully you can.
I can, yes. So please take it away.
Wonderful. Thank you so much. And thanks for the opportunity to present today on this webinar. We've heard a lot about adult cancers, and we're going to shift gears here for the last talk, focusing on precision medicine, molecular diagnostics, and pediatric solid tissue malignancies. So just a few background pieces of information about pediatric cancers if you're not aware. Pediatric cancer is the leading cause of death by disease in the developed world in children over 1 year of age. Although most children diagnosed with cancer have acute lymphoblastic leukemias, in fact, brain cancer is the leading cause of death by cancer in pediatric patients. And we know from large-scale genomics studies, such as the ones that were mentioned previously, as well as others, that pediatric cancers are very different than adult cancers. There are a few examples shown in the figures on this slide. On the left-hand side, we see a figure from a paper published in 2018 from the German group at the DKFZ, showing the small overlap between significantly mutated genes identified in thousands of adult cancers compared to thousands of pediatric cancers. A small number of genes shown below in the gray bubble, for which there are, in some cases, targeted therapies, but in many cases, these are either so far undruggable or tumor suppressor genes. And so we know from these studies of DNA profiling that a number of things are true for pediatric cancers. We really, for example, need to both profile tumor DNA as well as normal DNA comparators, so non-disease affected normal DNA, typically from white blood cells, are really both needed for study. Why is that in comparison to adults who are typically only profiled in the somatic DNA from the tumor? In kids, we not only need to identify these somatic mutations as well as insertions or deletions and amplifications on chromosomes, but we also need to look across the chromosomal arms, because in some particular diagnoses, those turn out to be prognostic indicators. So they indicate a risk of poor or good outcome. And we can act on that information in terms of the therapeutic aggressiveness or a decrease in therapy aggressiveness according to what the prognostic markers tell us. Of course, normal DNA needs to be profiled not only for the comparison to tumor DNA, but because kids, based on what we know about cancer, which is a disease of old age, should not develop cancer. And so in children, genetic susceptibility, which can be detected through the normal DNA optimally, is really an important characteristic to understand, and increasingly is leading to situations where, such as high tumor mutation burden, where the use of checkpoint blockade therapy may be used, and you've heard a little bit about that in the adult setting today. And then more recently, looking at methylation marks on the tumor DNA become important. They tell us about the cell of origin for that tumor and often add diagnostic precision that isn't there just simply looking at sequence data. Tumor DNA methylation in particular for brain cancers since 2021 has been part of the WHO guidelines for diagnosis and is considered to be a required and important part of precision diagnosis using a methylation microarrays with a classifier that groups the marks of methylation and helps to determine the tissue of origin for that cancer and therefore add precision to the diagnosis. And then finally and importantly, as illustrated on the right-hand side of the slide here, although there are fusion drivers in adult cancers, they are more numerous and different in pediatric cancers. And so getting a precision diagnosis, and as I'll show in a few minutes, getting actually some information about fusions that may be targetable with small molecule inhibitors or other types of very precise drugs is important in pediatric cancers. And because we can find these most readily looking at RNA, we also profile the tumor RNA in the course of precision diagnosis, as I'll talk about today. So targeting fusion drivers, I just mentioned, and in the few overlaps between adult and pediatric fusion drivers, such as NTRK fusions rather, as we can see here from clinical trial reports of different drugs that target NTRK and related fusions, we can see a tremendous amount of efficacy in these children who were included onto adult clinical trials, and in many cases have very durable responses with low side effect profiles, often for very poor outcome tumors such as these H3K27 mutated diffuse midline gliomas shown here in the upper right-hand corner. So if we can find a target in the fusions and there's a drug, we often get a high degree of efficacy for these children. We also have a lot of opportunities in rare cancers, and of course, pediatric cancer is writ large a rare disease, but there are certainly subsets such as papillary craniopharyngiomas studied here in a recent article in the New England, showing that because these particular cancers are driven by BRAF V600 mutations, which we understand very well from adult melanoma and other tumor types, and for which there are a variety of small molecule therapies, we can actually combine RAF and MEK inhibition here, as was done in this very small clinical trial of 16 patients, to find a very good response. 15 of 16 had a durable objective partial response or better, with a median reduction of 91% in tumor volume. So significant responses in these patients with very rare tumors and very long progression-free survival intervals, as you can see, and of course, minimal toxicity. And so this is really the paradigm. And in cancers such as papillary cranios, which do not have really an accepted standard of care, one can often intervene with a drug such as this and get dramatic results. And then finally, as I mentioned, there are a subset of children with very high mutation load cancers, primarily due to homologous repair defects such as CMMRD, constitutional mismatch repair, which is really indicative of, in adults, of response to PD-1 inhibition, and the same is true in children. And so these are two studies that were published in 23, really showing that there is efficacy for these children, often with brain cancers, but other cancers occur in this setting, and they do get, again, very good responses and highly durable responses as well, with a little bit of a difference in terms of the time to response, which is pointed out in these studies if you're interested. So I joined and opened the Institute for Genomic Medicine here at Nationwide Children's Hospital in 2016. And one of the things that we wanted to do immediately was to begin studying the children in our cancer care setting here at the hospital using this multiplex molecular profiling approach, which I'll describe for you here. We opened an IRB-approved research protocol in 2018, enrolled our first patient then, and closed this in 2022 because all of the tests that we were using essentially turned into orderable clinical tests in our clinical laboratory. So in this IRB-approved study, what we were doing was studying our patients with rare relapsed or treatment refractory cancers. We also included a small subset of children with rare hematologic diseases in this study. And the assays that were used are outlined here. So firstly, we used a tumor normal exome approach. So exome is obtained by pulling down the coding regions for every protein in the human genome. This is about 1.5% of the genome and gives you the ability to generate quite high coverage. Here we focused on attempting at least a 250-fold coverage depth on the tumor and normal DNA. That tells us a lot of information about heterogeneity in the pediatric cancer setting. But I'm calling this assay enhanced because in addition to the probes that correspond to the exons of the known protein coding genes, we also tiled the chromosome arms in the genome so that we could get a pull down at about 50 kb resolution to tell us about copy number changes that I mentioned earlier that can provide important prognostic information, and can also tell us about quite large deletions or amplifications around known cancer driver genes or genetic susceptibility as well. So this was one test that was performed for each of the children on this protocol. We also performed RNA-seq from RNA extracted from the tumor, and we performed multiple analyses using the RNA-seq data, but gene fusion detection was the primary focus for this assay. As I mentioned already, DNA methylation array data is important in the CNS setting, and so we implemented the Illumina EPIC 850K DNA methylation array and used the available classifier from the DKFC group in Heidelberg, Germany. Ultimately, all of the data for these samples, these patients, the results, implementation of any therapy that was indicated by our studies, where if we found a druggable target, we could reflex that into our clinical laboratory, provide a confirmation in the clinical setting, and then that information could be written into the patient's medical record for use in their care. And this all was captured within a REDCap database so that over time we could study these children longitudinally, but also put together reports of what we had identified, and those have been coming out over the last couple of years. Case reports as well as cohort reports, I should be clear. Here's just an infographic for the results of this trial. In aggregate, almost 500 patients were approved. Fewer were consented, about a 90% yield on consent. And then you can see other aspects in terms of sex, ethnicity, race, average age, etc. If you look over here on the right-hand side, you'll notice that the largest number of patients enrolled were CNS malignancies. And you'll also notice that non-cancer type here, vascular malformations and overgrowth syndromes accrued a quite large number of patients onto this onto this protocol. Here, if you're not familiar with vascular malformations and overgrowth syndromes, these are cases where cancer genes, typically PI3 kinase or mTOR pathway genes, have a somatic mosaic mutation profile. So only a subset of cells, for example, in a foot or an arm with overgrowth syndrome carry a pathogenic mutation in one of these genes, but it's not present in every cell. And the result of this is that very high-depth sequencing, such as I talked about earlier, has the sensitivity to detect these somatic mosaic mutations. They're often below 1% VAF. And if you can identify those, these patients now can take orally bioavailable FDA-approved drugs such as PI3 kinase inhibitors or mTOR inhibitors, experience a much slower rate of overgrowth, and experience a much improved quality of life. And so this was yet another way to use this type of analysis to identify these druggable targets in these individuals. The results of the cancer protocol are shown here really in terms of what we call medically relevant findings. So they're diagnostic, prognostic, or therapeutically relevant information, as I said, that could be clinically confirmed and provided into the patient's medical record. And they span everything from the germline cancer predisposition, which I told you about, to various targets such as somatic fusions, copy number variants, or somatic variants, as well as potentially targetable expression. Each one of these person-shaped blobs is the number of patients that were identified with the specific type of alteration, and then the DNA symbol over here on the other side is the total number of these types of variants that were identified. So in aggregate, with over 370 cancer patients studied using this protocol, over 90% receive at least one medically meaningful result that helps in this variety of the care spectrum. And we think that this really benefits, in particular, patients with rare cancers, as I mentioned a few minutes ago, and in cases where we had CNS malignancies studied. And the indeterminate diagnosis from pathology is in the range of 10%. We were, in many cases, able to solidify the diagnosis, which can also help determine the therapeutic approach. So, as I mentioned, we turned these tests, such as the enhanced exome and the methylation array, into clinically validated tests that we could offer to our patient population and providers. We also had already in place a commercial platform for fusion detection, which was the Archer Fusion Plex assay, and that had already been clinically validated. And so these three tests were then transitioned not only to the availability for ordering by our own practice group here at Nationwide Children's Hospital, but rolled up into a program called the NCI Molecular Characterization Initiative, which opened in March of 2022. And so I'll spend the bulk of the rest of the time here just talking a little bit about the MCI, the Molecular Characterization Initiative, and its impact on pediatric cancer. So this initiative, MCI, is a contracted mechanism that is supported by the National Cancer Institute of the NIH, and is a collaboration between our lab, the Biopathology Center at Nationwide Children's Hospital, and Children's Oncology Group. COG is the NCI-supported cooperative group that services pediatric patients across 226 different sites in the United States predominantly, although there are sites in Canada, Australia, and New Zealand as well that are affiliated with the COG. This infographic just sort of shows the workflow here. You can see that we have samples that will be coming in from all COG sites all over into our biopathology center, where they are evaluated for pathology imaging. The DNA and RNA are extracted and quality controlled, and then all that analytes and the case information come to our laboratory by courier. All of the case manifest and associated information for diagnosis on the patient is brought in electronically into our laboratory information management system, and the analytes are checked in or accessioned so that we can track them through the process also in the LIMS. These then obviously go into a series of laboratory operations to perform the three tests that I already described. The data are analyzed in terms of the bioinformatics analysis in the Amazon Web Services cloud that we have as a private HIPAA-compliant cloud. And the analysis, interpretation, and the clinical reporting takes place in our clinical group with our variant scientists and clinical directors. Once they sign out these reports in our laboratory information management system, the reports automatically go to the providers using data delivery through Amazon Web Services. So this essentially notifies the provider that they have a result. They can use their CTEP secure sign-on to access the portal and download the results for that patient in a HIPAA-compliant environment. So this clinical testing that I'm mentioning actually gets completed within 14-day turnaround time. We really want this to be clinically relevant for these patients, whether it involves identifying FDA-approved therapy, whether it identifies a prognostic marker, whether it gets them onto an active accruing clinical trial or what have you. And so really 14 days turnaround time in our clinical lab, 5 days turnaround time at the BPC to turn the tissues into DNA and RNA is really the total time frame that we're talking about here. Importantly, although we're delivering this clinical information to children across the United States and in other countries in a very rapid time frame, we're also helping children and with pediatric cancer into the future. And as you can see in this last bullet point here, once the Amazon Web Services data delivery engine goes to providing this report to the provider, we also use our web services tools to auto-redact all of the PHI, the private identifiers in the data, and then Amazon delivers this into an NIH repository, which is the CCD, the Childhood Cancer Database. So this database is really meant to house these data, individual tests from a patient, as well as their pathology images, as well as any ensuing clinical data, et cetera, will all be present and linked together with a universal identifier that, of course, does not identify the patient. But the bottom line is here that we're also perpetuating research in pediatric cancer into the future. And that is, I think, critically important. So just quickly here, an overview of the analytics. We have all of these Dockers present in our Amazon Web Services cloud, and you can see that the different assays, tumor normal exome, tumor RNA for fusions and ITDs, and tumor DNA for methylation, all are analyzed in the Amazon cloud. The clinical reports are signed out, and then these are the different types of results that can be put into the clinical report for each child. All of these assays are validated not only in CAP/CLIA, but also New York State Department of Health, because we do have patients from New York coming through this testing as well. So this has been a fine-tuned engine, but every once in a while there's a spanner that gets thrown in, and that happened to us a year ago or so, where Illumina came to us and others that were using the EPIC methyl arrays and said, we aren't going to be offering these anymore. We're changing them to a 935K version 2 methyl array. And at the same time, we learned from the DKFC that the corresponding classifier they were developing for the version 2 arrays would be part of a commercial offering. And, we were welcome to upload data to their web portal, but that doesn't really work in a regulatory environment. You can't have your analytics somewhere where you have no version control over them. So we went ahead and developed our own classifier using publicly available data. And this has a silly name, MacDaddy, but it actually stands for all of these words: methylation array classification, diagnostic, and data integration. And this is being used for the precision diagnosis of central nervous system tumors. So the top 2 bullets just repeat what I already said about the situation with the new arrays. We do have some differences here in terms of people who might be interested in the nitty-gritty details. DKFC classifier uses random forest. We actually used a regularized generalized linear model, machine learning approach to train the classifier. And essentially, we trained this on both version 1 and version 2 array data, and so it's compatible with both array formats. This was validated in our clinical laboratory and went live in May of 2024, and we've been using it ever since, and I'll show you some data for that. And kudos to Ki Chin, who's our machine learning expert in the group, and Marco Leung, our clinical director, who oversaw the validation for these arrays. Here's just a few quick facts about how we did this. So I mentioned already that we had many original data sets from publicly available data and classifiers that had happened in the past. This is the IDAT format that comes through the methylation arrays. And this perpetuated a training data set from the DKFC that went through our first modeling step here with 5-fold cross-validation, and then a multinomial RGLM second go round with another cross-validation to finally output the classifier results. Classifier results come out as illustrated as named rather superfamily, family, class, and subclass to provide increasing levels of precision about that type of tumor with a corresponding score that gives you a confidence in whether how confident the caller is that the designations for these 4 different areas are correct. And so the bottom line is that we were able to get all this data, do the training, and use the same 4-level classifications as the DKFZ classifier so that the reports would look familiar to our recipients of the reports. So this is just a few facts about the training and validation performance. As you can see, in general, we had high levels of concordance using the online version 12.8 classifier with DKFZ. We had a small number of samples that could not be accurately diagnosed by either platform, which is not surprising. And we had some differences between the two in terms of a DKFZ classifier having a scorable result and our classifier not, and vice versa. The bottom line is that we are largely concordant with the DKFZ classifier, as I mentioned, in a few settings where we don't necessarily have enough samples to really train the classifier well enough to give a subclass score. We actually just default to some of the WHO designations rather than to try and slice and dice it even more. In those situations, that sort of granularity wouldn't influence how the patient was treated anyway. So just to summarize then, this is what a report looks like from the methylation classifier. You can see here the superfamily, family, class, and subclass designations and the corresponding scores here, where confidence sort of ends at a 0.8 designation with a diffuse glioma with a mutation in the IDH gene. We also report out clinically the MGMT methylation status. This determines whether temozolomide can or cannot be used based on prior studies. And we provide 2 research results, a copy number variation profile, because all of these mutations across these sites across the genome for CPG can give us good copy number information. In addition, we plot out using a Tisney approach, the overall similarity between the subject under study and a very large database of already classified pediatric brain cancers. And then we also have a localized plot that's interactive where the clinical director can zoom in and really see the other tumor types that are present in and around that individual's result. The copy number variation profile also can be compared to the copy number profile coming from the enhanced exome sequencing. And so there is this integration of data that can happen as you go along. And indeed, some of the methylation classes, this one is just a mutation, but some methylation classes actually will report out on the prediction of a driver fusion being present in that tumor type as well. So this is just a quick zoom out on this patient shown here in red, which is grouping with a series of high-grade pediatric gliomas. And so the MD can look at that for some additional information about the grouping of that patient based on methylation. And then this is just a whole genome copy number plot going from chromosome 1 here on the left to 22 on the right, where you can see some significant deletions of chromosome arms and an amplification here on 1q. So just to wrap this up so far to date, as I may have forgotten to mention, we've now studied over 4,800 individuals through the MCI since March of 22, so getting close to 3 years on this 5-year project. The methylation array results, as you can see, we can call out with high confidence about 86% of these individuals using the methylation array. And again, that information couples with conventional pathology information. We're now working on developing a sarcoma classifier based on methylation. And I mentioned about 10% of brain cancers can't be accurately diagnosed just by pathology alone. That number is closer to 25% for some sarcomas in the pediatric setting. So we think this will be helpful. Here's just an aggregate look across the United States as of March 1st. As I mentioned, 4,800 patients enrolled and consented onto this MCI. Of the COG-affiliated institutions worldwide, we have samples, at least one sample from 206 of those 226 institutions. You can see the number of orders received where every patient equals 3 orders. About 90% of the orders can get signed out within the 14-day turnaround time. There are a variety of things that influence that, and I'll talk about those in a minute. And so far, we have patients from 47 states, Washington, D.C., Puerto Rico, and about 10% of the tests are performed for international patients in Canada, Australia, and New Zealand. We're adding additional tumor types. To date, we have studied CNS malignancies, soft tissue sarcomas, a very large number of what are called rare cancers that can occur in many sites, and neuroblastomas. And next up will be Ewing sarcoma that should open sometime in April of this year. In addition, we're transitioning away from the use of the Archer platform to get a broader characterization of RNA for each patient, especially important for the data going into the childhood cancer database. We think this is a much richer source of mineable information than just knowing what fusion was detected. And so we're working on that validation clinically actively as we speak. Here's just this quick look at COG institutional enrollment on MCI. So about 50% of the enrollments come from about 35 institutions, most of them tertiary care hospitals for pediatric patients, and about 50% from another 173 institutions that have submitted at least one patient onto this protocol. Here's just a look at enrollment by age, where we can enroll between ages 0 and 25. So we're hitting that spectrum pretty well for soft tissue sarcomas and rares. Neuroblastoma is an early childhood cancer, so no surprises there, and CNS malignancy sort of the same. And then lastly, you know, this didn't start out sort of at the numbers that we're looking at here, but since the opening in March of 22 on the left, we have come up rather quickly over time in this 3-year time period to where we're now routinely receiving about 200 cases per month for our clinical testing. Here's just a quick look at the aggregate of what we see in terms of germline and somatic testing. So you can see the different disease groups listed here that I already told you about, the relative number of cases. This is as end of January 25. And then you can see proportionally germline single nucleotide variants, germline copy number variants, and then the total cases with a germline finding. I'll just say for reference that most large-scale pediatric cancer studies in the past have found about an 8 to 14% range of germline susceptibility. You can see it's about 25% in this group of rare cancers that I mentioned, which is a very broad group. But I would say that even with next generation sequencing with exome coverage, we're probably still missing some significant amount of germline predisposition that will likely take things like long read single molecule sequencing to add more granularity. But in total here, about 14% of patients with a reportable germline finding. And then the somatic findings are much higher, predictably, and range as high as almost 94% here in neuroblastomas, between the combination of point mutations and copy number variants. For solid tumor fusions, you can see also the results here in terms of the proportionality on the right-hand side of positive results. I should point out that these are not just fusions, but also include a small subset of changes to protein kinase genes called ITDs, or internal tandem duplications, which are also driver alterations, and we can pick these up as well. And so those are included in those numbers. And just to wrap this up, there was a really nice paper recently published in JAMA Oncology. It's called a review, but it's actually really a position paper that advocates that at least in the CNS setting, as I mentioned, these 3 tests really are needed to get the full granularity and precision on diagnosis and result in best outcomes for children. this review actually argues both from adult and child perspective that doing more comprehensive testing, including the methylation, broad DNA, tumor normal comparison, as well as RNA, is really now merited in this very complex set of cancers that occur in the brain. And in particular, noting that the aggregate cost of all this testing, if you look at the aggregate cost overall of the care for individuals with brain cancers, is about 2% of the total hospital bill. So really arguing for insurance companies to begin reimbursing these tests in a more appropriate way. I'm just going to spend the last 5 minutes here talking about not all of these challenges, but just the last two. But what really keeps us from doing a 14-day turnaround time? And in particular, one of the challenges is just that about 60% of the samples that we receive come from formalin-fixed tumor DNA, which does terrible things to tumor DNA and RNA as well. And in particular, some of these come as just sort of small numbers of needle biopsies. And so we're not only challenged by poor quality DNA from formalin, but we're also challenged by just not very much DNA being available. And keep in mind, the tumor DNA has to go to methylation testing as well as to the tumor normal exome. And then the last, I think, is probably familiar to most people, and that's the ability to scale is really challenged by the complexity of these tests and the desire, of course, to sign them out with high degree of accuracy and not miss anything that we return to our patients. So just one quick slide here on how to get around the poor quality, low input. On the left-hand side, you'll see that our current clinically validated assay for tumor normal using next generation sequencing uses an NEB library, which is a fairly conventional approach to putting adapters on the end of each fragment. But when you look at the fragment population that can really be amenable to this, it's a small proportion. Perfect and slightly damaged, but repairable. So that limits the complexity of the library, which means that for formalin-fixed material, as you can see, we have to put in 500 nanograms of DNA just to get a good quality library. And sometimes that's not even enough. What we've moved to and have recently clinically validated is an approach from a small company in Santa Cruz, Claret. And Claret essentially requires only 25 nanograms of input for formalin-fixed DNA or for frozen. And that's because the first step in Claret, as you may be able to see here, is essentially that we fragment and denature the DNA. So everything starts out as single-stranded DNA. And so everything is treated equally with respect to the second step, and that's adapter ligation. As a result, we can have a very small input and still get a highly complex library that's suitable for the high coverage that I mentioned. So we're very excited about implementing this, and it has a go live that's coming up here soon in our clinical laboratory. Finally, and not surprisingly, human evaluation is a huge bottleneck to throughput. This is just a graphic that sort of illustrates, if you will, that lots and lots of databases have to be consulted, as well as reports from the literature, et cetera, to really get a comprehensive clinical report and sign out for these patients. And so we have two things going on here that are going to become intertwined. One is the variant categorization tool, or VARCAT, as we call it. This is actually starting today. Will be clinically validated in our lab, and essentially the notion behind VARCAT is twofold. One is to harmonize the way that variants are described, so that using VARCAT anywhere in the world, you can have a similarity of terms that are being used to describe any variant, using a variant classification framework that's largely been outlined by the VIC, the Variants in Cancer Consortium, which is very focused on harmonization of report outs for clinical data from cancers. And Alex Wagner, who's a PI at IGM, and Wes Gore in his lab have really been the drivers for VARCAT development and implementation here towards clinical validation. Then VARCAT will become part of another tool, which is generally a group of tools called Automated Variant Analysis Toolkit. this sort of describes what's happening here, but it's basically an attempt to streamline the process of evaluating all of these variants. It also stores inversions and can retrieve existing data that we've seen before so that we can annotate those pieces of information into new clinical reports for new patients. And then it also has a very clever approach here in terms of the aggregation of information so that it's all right in front of our clinical directors. Finally, within our lab's information management system, we have a shopping cart feature that is allowing the clinical team to sort of buy the variants that they plan to report in the clinical report. And once those are all bought for germline and somatic, for example, then they buy what's in their cart, and those pieces of information along with the variants can be automatically written into the clinical report. So you get this amazing amount of reproducibility from person to person, but also from case to case that goes along for the ride. So this is just a look at the system that will be AVA overall. That includes these various databases that I sort of alluded to earlier. In one part, the variant categorizer, a visualization tool for where variants fall in the gene itself, the data warehouse that underlies all of this and captures the information, which is our Varhouse 2, and then our LabVantage is our LIMS system that allows us to interact in writing reports and issuing reports. And then finally, and importantly, we want to share variants and information with the outside world as well, with ClinVar, within our EPIC system for clinical record keeping for patients, and so on. And so really, perpetuate our long-held position of being data sharers and tool sharers. So just to conclude, I hope that I've convinced you that these clinical genomic assays really add important clinical evidence. They can impact treatment planning, diagnosis, and prognosis. And unlike adults, we really need to combine DNA and RNA results to give us the full picture that's present here for fusion drivers, cancer drivers, genetic susceptibility, and methylation-based classification. This is truly critical. And we're scaling our molecular profiling efforts and hoping to offer many of these tools to the outside world to enhance the cross-disciplinary accuracy in these variants and how we understand them over time. So I'll just finish by thanking everyone. It takes a huge effort, and I don't want to call out anybody's name in particular, but acknowledge the collaboration with COG and with the National Cancer Institute, and with our BPC colleagues here at Nationwide Children's Hospital, and really acknowledge a huge amount of effort that takes to put this all together every day for kids in the US and elsewhere. Thanks for your attention.
Thank you very much, Dr. Martis, for that wonderful presentation. So let's get to our final Q&A session of the day. And I just want to remind our audience that you can still ask questions at any point using the Q&A tab on the right-hand side of your screen. Dr. Martis, you mentioned that FFPE samples are probably your biggest logistical obstacle. And you mentioned that there were some ways to get around it, and I think that a lot of people are kind of developing workarounds or ways to better access FFPE materials. Is there any impetus on the clinical side to move away from FFPE just as a primary sample storage vehicle, or is there no alternative on that front?
It's a great question. A lot of conventional pathology revolves around the preservation of tissue architecture, and really nothing does that better than the fixation with formalin and embedding in paraffin. So I think rather than try and fight it, we have really just searched long and hard for ways to get around it. And, to some extent, it's very dependent upon the situation. So I don't want to just say that, pathologists are resistant to change. In some settings, such as a large brain tumor, for example, where the whole tumor is removed at surgery and you have a very large tumor mass, you have the luxury of asking your neuropathologist to set aside a frozen piece and maybe also adjacent to the piece that's going to be used for the for the conventional H&E evaluation or what have you. More frequently, however, there are less invasive ways of sampling for diagnosis, which is often what we're talking about here, or especially in situations where the tumor can't be resected safely. And there are many situations like that. What one is left with is essentially using a biopsy needle to sample into the tumor. Typically, several passes of that biopsy needle are performed, where the tissue that results from each pass, gets put into a cassette. So now you're talking about, 10 micron biopsies, very small, that don't really have a lot of material. And now we have to sort of share between conventional pathology and all the molecular diagnostics that we want to do. So I think with or without formalin, we're still in this setting where sometimes we just don't have the luxury of differential preservation to suit all of the things that need to be done to get us an accurate diagnosis. So that's kind of where we're at from a accept it and move on standpoint, I guess.
Is it also the case, you would say, that pathology is the first priority for any sample, and then genomics or other molecular diagnostic techniques will be the secondary concern?
Yes,
that change in the future?
Yeah, it's a great question. I mean, I think the answer clearly is yes. I think, molecular pathology is a very interesting place right now because there is this sort of emergent collision, if you will, and I don't mean that in a bad way, but between sort of more cutting edge genomic technologies like digital spatial profiling that somewhat intersect the conventional pathology realm, right? We're sort of looking at tissues. Now we're able to use formalin fixed material, curiously enough and happily to do these assays, but we're often dependent upon, it's essentially sort of the new version of immunohistochemistry, right? Where you have an antibody that's labeled, and if it's labeled with an oligo, then you're reading it out on your next generation sequencer. But oh, by the way, you can also do the transcriptome while you're at it and see what the signaling is that's going on, and so on and so forth. So I think it's a really interesting question now in terms of kind of, yes, we need conventional pathology. We really appreciate and value the way that pathologists are trained and the way they think about diagnostics. But it's also just really very much in flux in terms of these new technologies and how and where they actually fit. And will they, for example, ever be as cheap as an H&E to perform, which is probably the most important question, but I won't go into that.
Thank you very much. I think just to wrap up our session, I just want to ask, you noted that you had about 50% enrollment in terms of data. What's the word I'm looking for? Data submission from about 35 institutions, and then the rest came from about 130 different institutions. Is that correct?
So that was not data. Those were children, the percentage of children enrolled. Yeah.
Is that kind of to be expected as kind of a product of just population density clustering, or is that a potential risk of oversampling certain areas?
It's a good question. I think it has to do with a multitude of things, but the factors that come to mind right away are patient volume. So obviously the large tertiary care hospitals in the US have much higher patient volume, whereas some of these patients in smaller states, maybe even being diagnosed in the community hospital setting, so not a dedicated pediatric oncology setting. The other obvious barrier here is just a little bit on the side of, wow, this sounds really great. The testing's being supported by the government, but how am I ever going to interpret this information to make sense for my patient? And I think that is really where COG has stepped up. They've sort of provided office hours from, more experienced, molecularly savvy oncologists that can help address questions for providers that are just not sure what to do with the data. And that includes the germline susceptibility data as well, which can often, as you probably know, invoke things like family testing and determining whether there are siblings that also have similar cancer predisposition, et cetera. So I think that's how it's been addressed, but I think those are probably the two biggest factors that come in there.
Thank you very much for that response and for the presentation. Thank you for being here with us today.
Thank you.
So just to wrap up, I'd like to give our audience a final reminder that the symposium will be archived on the scientist's website, and you will receive an email notifying you when the on-demand recording becomes available for viewing. Please also make sure to follow the scientist on social media for updates on other events in the future. In closing, I'd like to thank everyone who took the time to join us today and extend a final thanks to our panel of speakers, David Liu, Muda Barbiro, Omid Visse, and Elaine Mardis, as well as our sponsors, Biomodo, Epicipher, Biorad, Sinobiological, Biodium, BD, BioXcel, Scale Biosciences, and Selecta. Thank you, everyone, and have a pleasant day.